Zapli to chodí na ty. Já jsem zapnul tu klidu, tady už je nedýchatelnou teď. Tak já nevím, jestli zavřít ty dveře. Asi, že kupak větra.
Asi budeme větrat mezi.
Máš teďkom start, 1 za hodinu, ale hodinu, jo, vlastně ty budeš plynule mluvit rovnou do toho track designu. A máš to celou hodinu? No ne, víš, nevím, jak to tady udělat s tím větrák. Já zkusím.
Pověř holky, jak to vyřeší. Well, people online.
Vyřešíme v průběhu, jo? A to budeme počít. Klidně v podstatě za minutu můžeš začít. Jo? Začít mluvit, když se to funguje.
Okay, ladies and gentlemen, welcome here in Olomouc to the 6th version of Advanced in Silico Drug Design Workshop Challenge. As you can see, there is plenty of us who will be speaking to you, trying to give you an update about what is going on in the in silico drug design world, and trying to teach you also some tutorials about how some methods can be used by yourself. We are here at the Palacký University, and you might not know that this is actually the first and oldest university in Morava. And that's especially saying to the people from Brno, because they are saying that their university seems to be also quite old. But we have currently this year, we have actually 450 years of this university. And as such, we welcome you here at this really interesting and really nice university city. As you will see tomorrow, hopefully we will not be with too much snow and too freezing temperatures. But after the freezing temperatures, we will be arriving here at Moritz Brewery, where we will have a workshop dinner tomorrow. So that's something to warm us up. Speaking about the workshop dinner, here is the program of the current version of workshop. Today, it will be basically a basics about where to find the data, and we will have a guest from the ChEMBL, from ChEMBL EBI, who will be speaking to us via Zoom. So hopefully, fingers crossed that this will work. And unfortunately, the rest of the today is with me, so sorry about that. But tomorrow, we will have the guests speaking about mainly the pharmacophores and metabolism and natural compounds. On Wednesday, it's basically on a group of Pavel Polishchuk, major co-organizer of this meeting, and it will be about the ligand-based methods. And on Thursday, it will be, again, a lot of on me, but thankfully not only on me, because there will be membrane dragability via molecular dynamic simulation by Semeny Esilevsky, and also high-throughput molecular dynamics by Alexandra Ivanova. And on Friday, we will have a challenge, which will be a ligand selection, if I'm right. And you can win some nice prizes on that.
I hope so.
Yeah. And one of the prizes for students is that you can win 4 ECTC credits. And that's for everybody who will be participating. So don't worry about if you will not find any one molecule, that's fine with us. Not much with the pharma companies afterwards. Speaking about the materials, all materials, once we will be showing it here, all PDFs will be put similarly as book of abstract to our webpage. You are probably familiar with. All of these lectures days will have a Zoom link. So if you are ill, you can look at it from your hotel. And also, smile, we are on YouTube, currently broadcasting. So with that, that's it from me. Do you have any questions? If not, enjoy this workshop. So I'll quickly switch.
You can switch. I need this microphone. I hope that participants will hear me, so I will just introduce myself. So my name is Pavel Polishchuk. I'm as the head of the group of cheminformatics and drug design in Institute of Molecular Translational Medicine. I welcome you here in our snowy Olomouc. I hope you will enjoy this workshop. I would like to remind about the excursion to our institution. So who are interested to look on how our institution staffed, because our institution mainly work on anti-cancer drug discovery. We have high throughput screening pipelines. We have animal facilities. We have DNA labs and so on. So if you are interested in all this stuff, you can put your name in the list of participants, and on Thursday, you are welcome to visit our institution. So that's all from my side. So enjoy this workshop. Thank you.
Okay. So in the program for this time, there is the lecture about drug design introduction. And let's do it. In order to use some pharma to develop a new compounds, if you are an organic chemist, you usually cannot work without the NMR, because you need to find out what was something that you prepared. And you can think about the computational drug design as another level of the visualization and thinking of the compounds, because of course you can design compounds without drug design in rational way in computer, but it's sometimes better to use it, because some of these methods can help you to sift through a vast universe of compounds there is. So in this lecture, I will be speaking shortly about sources of drugs and what are actually the drugs which were approved last year. And I will be speaking about the drug designing problem and a little bit differences, because you will see that the currently small molecule drugs is making like 2/3 of the drugs, but not all of them. So let's see. So the first drug, which was taken from the and purified, was this nice compound called morphine in 200 years ago. And it was used for a really nice ways, for instance, as a way how to put babies asleep, which as a father of two, I would use probably sometimes in this. But following this, usually when you have a drug, we all know that the drug is taken from the toxin basically by the dose. And therefore, to find the right dose that can cure people, you have to test somehow what dose it should be. And here is how the people tested. So at the beginning, it was tested on humans. Later on, it was tested on prisoners. Actually, the first clinical trial, which was done by the English Marine, was done on English mariners, of course. Yeah? And it was about the usage of the citrus plants in order to cure fifth of falling. Then, well, trying anything on humans is kind of a tricky thing still. So it's better to use animals. Well, not much these days. So it's better to use the isolated organs taken, let's say, after the animal was killed in order to get the meat. You can use the rest of organs to test the drugs. But it's not really useful in a large manner. So slowly we move toward the cell lines and large chips and rationalized high throughput screenings and so on, usually on enzymes, cell lines, and so on. So these days we have high throughput libraries that are sift through millions of compounds by robots to trying to find out the active compounds. And in silico way, we are basically doing the same thing, but without the need for having the cell line and without the need for having a real physical manner other than the cloud computers. So usually our compounds are natural derived, which makes sense because we are natural beings. And therefore we are basically mimicking quite a lot the drugs or compounds that are present naturally in our organisms in order to cure. But you can see that completely synthetics are still making about 30% of the drugs at all. So since we are speaking about this topic, it's probably useful to set up some kind of vocabulary. And the major vocabulary which we need is to distinguish between the target, which is the biomolecule we want to hit with the molecule of the drug, the lead, which is a base molecular motif that actually can be derived afterwards to making by optimization towards the final drug, a hit like a compound which you have some kind of screening, and you set up a threshold that will give you the information of compounds that will bind stronger, let's say, or be active enough to show the hit. And from the hits, you select the candidate compounds, and you test them for the efficacy, which is basically qualitative, whether they work or not, and activity, usually measured, because usually the compounds are developed as inhibitors, therefore as an inhibition concentration of 50%. And then the amount of the drug which you need is quite depending on the drug and the target to find out, and the lowest is usually the better. But of course, the compound has to go where it needs to go, so you have to also think about the bioavailability and possibility of drug metabolism. So how are the drugs usually approved? Conventional drug approval is that first you run some preclinical testing, then you have which is basically in lab and later on animals. From the animals, you move towards the phase 1 with dozens of participants to test basically the safety of the drug. Then you test in phase 2 by hundreds of participants, the safety and immunogenicity. And then you go towards the thousands and participants, safety and efficacy. And this is on vaccines, but for the drug, it's similar. You are just looking for the individual endpoints for those studies. And then after that, you get the registration and finally approval, if you are lucky. And in silico drug design can help you basically mainly in the first version prepare the molecules that would be successful in that part. But we are behind. Well, after the first few waves of the pandemic of COVID-19. Currently, I think it's the wave like wave 6 or something, depending on which country you are in, because the individual countries have different waves and different problems with them. But we've seen during the pandemic that sometimes the things can be done quicker and more time efficient. And this is the way how it's done. So first you basically collate the things, and even if you have first results that seems to be going in the right direction, you move towards the phase 1. You often combine the phase 1 with phase 2. Because if there is need, you basically enroll a lot of people, and therefore you can move towards the phase 2, and you can combine it with phase 3, and really quickly within 1 or 2 years, in comparison to, let's say, 10 years, you get the approval, but only a provisional one. We'll see later how much of the drugs is actually going through this quicker approval rates, not necessary within 2 years, but within, let's say, a quicker period. And then you have the safety monitoring afterwards, pharmacovigilance, and in these cases, really strict one. And after that, you get the full registration. Which reminds me that there are several reasons why you can have some of those drugs move along quicker. One of the possibility is if you have something first in class. So if you have some kind of a disease for which there is no other option of cure. In that case, of course, your cure can help a lot of people earlier, and therefore these ones are usually moved. So most of those is rare diseases. And the rare diseases where you have an orphan drug, therefore there is not much people that would be taking the drug afterwards. And therefore you can skip a lot of the safety checkings for the larger populations, which speeds up the process. But of course, if it's rare disease and there is no cure for those people, it can be a life-changing event to have the drug that actually cures them, because previously there might haven't been any. Yeah. Then, if there are some serious life-threatening diseases, like the COVID one, then you can have a breakthrough one in order to get through quicker. And some accelerated one can be if you found out early enough that your efficacy is really good compared to the placebo or in comparison to the previous medicines, then you can basically shorten up that testing one, the testing season, because you've already shown on the smaller amount of people that it's working enough. What's going on here? I would like to really see. And if you are going through those, you usually have a conditional market approval, and that's just this pre-registration when you start still to have the full registrate just before the full registration. So how many drugs we actually have per year? Approximately 40 per year is either in US, where these amounts are usually taken into account, there are actually 2 drugs already registered for this year by FDA, and approximately 35 or 40 are registered by the European Medicinal Agency for human use. Actually, the first in class along these is usually approximately 40% of the drugs. And this is mainly those orphan drugs, and these orphan drugs are actually increasing by the years. When I was preparing these graphs yesterday, I was looking for the last 10-year periods and compared the previous first 5 years, 2010 till 2015, 16, and the later one. And you can see that those orphans actually increased in Europe from 30 to 50 and in US from 40 to 50. And those accelerated registrations increased also. In Europe, it's smaller, it's about 35%, and it basically fluctuates around this way. But in the US, they really sped it up from 60 to 70% of drugs are actually going through these expedite approvals. And the major reason is that waiting for those companies is really a financial problem, as you will see later. So what type of the drugs actually we have? As you've shown on the previous slide, it was 37 drugs which were approved in the last year by FDA in US. And those more compounds are small molecule ones, those without those surroundings, proteins, and the antibodies. And on the next slide, there is also some radioactive compound, which actually has a Czech origin, because that was developed by Martina Benešová, originally from the Charles University, now in Heidelberg. And there is quite interesting usage of the xenon compound that are used for imaging. So those are actually inorganic drugs. So we still can have them. But if you compare how many small molecules, organic compounds are developed or approved, it's basically the same amount as the biologic ones. So during these lectures, we'll be speaking mainly about the small molecules, apart from today, when I will be speaking about the large protein ones and biologicals. But unfortunately, it will be only a short briefing to those types of the development of the drugs, because as you can see, quite a lot of those is actually something that's missing. Or if you want to kill it by the antibody because it's more stronger than stronger binder than the small molecule one. And well, you might not know, but there is a problem in the drug development, which is called Ehrum's law. It's actually spelled backwards, the Moore's law, which you probably are more familiar with. And in Moore's law, basically the speed of our computer is speeding up every 2 years by twice or something like that. In Ehrum's law, we are backward, which means that actually the amount of money you need to have one drug is increasing and increasing. And that's mainly due to the problems with the testing and the problem that we basically are trying to gather the compounds which will be working as a Me Too drugs, which are quite problematic. And there was a paper recently in the review of financial studies by Krieger et al., where they've looked into the drug development in comparison to what type of the drugs were already registered. And by looking at the similarity to the previous drug. So here is the Tanimoto similarity they've used. You will be seeing that probably on Wednesday a lot, but probably tomorrow also. And this basically looks into the compounds and trying to find out whether they are similar with via their structures, via the features, and so on. And these are statins. And you can see that the first statin was lovastatin, which was approved in 1987. And the similarity score to the previous drugs was only 2.25. And everything below 0.6 basically is a quite different drug. But then the pravastatin and simvastatin, which were both registered in 1991, the Pravachol was similar to 0.61 to the previous one, and Zocor, simvastatin, even had a higher similarity score because it has the higher similarity more to the Pravachol. So you can see that the similarity of the compounds which were registered is increasing over time. And if we look into the distribution of those compounds that were, when they were registered, what was their similarity, there is a high peak around 1, which is simple explanation because that's a Me Too drug, which usually is deuterated. And there is actually a nice example of that just now, because you are probably familiar with remdesivir, which was the first drug which was used to cure the COVID-19. First, it was used in hospitals because it was put into the veins. But now it's actually added earlier because it was shown that it does not help much when you are already hospitalized, but it can prevent the hospitalization in 80% of patients. So now the usage of remdesivir after 2 years of the pandemic has changed it a little, but it's still used and it's still fully registered. But interestingly, there is a new drug which is called VV116 from Chinese company. And this one is similar, but it's deuterated, remdesivir. And as a deuterated, it goes around the patent, the original patent of remdesivir. So it's all okay to go. So these are the deuterated ones. Quite nice trick to do that, yeah. But these ants are the low one. But you can see that there is a large steep towards the similar, more and more similar compounds. And if we look into the how often these things changed and how often these drugs are actually approved, we can see that the likelihood that the FDA would approve a more similar drug to something that's already there is of course increasing. Yeah, which makes sense because if the drug is similar to the drug which is already approved, it will be probably similar in the effects, and therefore, if the previous drugs was already going through the approval, it's more likely that the new drug will pass as well. But if we look into the patent value on those drugs, it's actually decreasing. Because of course, if there is a drug already in the market, then the original drug will be, after some period, generic, and the new drug going into that same direction will be competing with this generic. And therefore, its price will be quite lower. If it's similar in the chemical mechanism, of course. So actually, it makes sense to look into the novel ones. But it's more risky because you had smaller amount of approval. But if you have the approval, then it goes on. And since it's riskier And since the drug development costs are going to the roof, over the roof, of course, you need a large company to do that. And if you compare how often those novel drugs are developed in the companies, depending on how many drugs they already have on market, it's actually quite nicely shown that if it's larger company, then they usually have the capital in order to push the drug through the registration process and to get the approval and to get the drug to the production. Then the small startup company with no previous drug. So contrary to the popular belief that the larger pharma companies are basically not doing novel drugs, they do more often than the startups. Just in order to give you the sense of a slightly different field, sometimes we do the drug development not on the medicinal drugs, but on different fields. For instance, in my case, I do a lot comparison with the plant scientists at the laboratory of plant regulators here in Olomouc. And so I've looked into the agrochemicals. What's the status there? And interestingly enough, we are thinking that 40 new drugs per year is not much, but in comparison to the agrochemicals, it took quite a while then they reached the market. So you can see that now in 2022, there was 14 in development, 0 yet approved of those. And out of those which were in 2017, only 5 were approved and 11 is still in development from that year. So it's still order of magnitude lower than the medicinal drugs. And the reason is there is even smaller amount of companies, and it's still not enough of the push to generate those agrochemicals as comparable to trying to find out the new cures and medicines. So, how we design? The problem with the drug design is that it's a hard problem. It's a hard and expensive problem. Because first, the expenditures for one drug is $1 trillion. And that's only the development, not counting for the productions, patents, distribution, all of those stuff that are also quite costly. And especially since the new drugs are usually larger and larger, their organic synthesis is more expensive as well. And therefore, the new drugs are quite expensive. This is probably too low for today. I've taken it when I started with lectures on drug development, and it was in 2011 by the Czech Agency for Drug Registration. And it's also a hard problem because it's not simple, not easy to find out the pair. So the expensive, we are on the cheapest part, basically, €3 per one compound. But of course, since we are cheap, we can have more of those compounds. And that starts to be more expensive if the price of electricity is going upwards these days, but happens. And if you are testing anything on something livable, then your price is going upwards as well. But it's not comparable to the testing on human beings and volunteers, because that's the most expensive part. So you really want to put there a compound that would survive and not fail at that stage. So you want to fail quickly if you want to fail. And it's a hard problem. Now we know the structures of all 27,321 human proteins, thanks to AlphaFold. But due to the alternative splicings, there can be a larger amount, and there is quite a lot of the RNA taking control and regulations and stuff. And usually the regulation of some biological process is something you want a drug for. So that's probably in these two parts, the RNA and the protein-protein interactions, that's probably the way how this will evolve. And even though we have 60,000 experimental structures for human proteins currently, it's only 12,000 that are unique. So experimentally, we have quite a lot of structures, and computationally, we have all. So this is new for the last 2 years that we have this AlphaFold database. But still, going from the protein towards the protein compound, it's quite problematic. And there are multiple possible obstacles that you can find out along the way. The first one, you don't have a testing model. Quite notorious is a problem of testing HIV, human immunodeficiency virus, on mice. Because you want to test it on mice, but in order to do that, basically need to make the human immunity and somehow insert it into the mice. And the biologists were trying to do that a long time and still did not succeed. So, problem. And sometimes it's quite problematic to test the drugs if they don't want to. In case of rare diseases, it can happen that there is just not enough people to pay for the drug development afterwards. If you have a really rare disease, which only thousands of people around the world has it, to develop something for them for 1 trillion of euros, probably not sensible investment, to put that bluntly in the money-wise. So, yeah. And that's actually the reason why the orphan drugs are easier for registration. And currently there is quite a lot of drug development which is focusing on orphan drugs as the first ones, and then to take the drug which was already successful for orphan, and then try to enlarge for the larger audience afterwards. Yeah, so that's a new stream about how, even with the smaller amount of money, to develop a drug that can have a matter, that could be really life-saving medicine, and then slowly to enlarge its use efficacy towards the more and more possibilities. The typical problem why currently drugs are not approved is that the previous drug is working better. And the new one is not toxic, but also not effective. So small effectivity is one of the biggest trouble these days. And another problem is active compounds may be already patented. So sometimes it's quite hard to find the way along the patents to basically get your drug into the market. So given the illness type, you have to also think about what probably can be used as a cure. So just two viruses, HIV versus COVID. For COVID, the first thing which we had was remdesivir, and the most used one was actually vaccines. For the HIV, there was a tremendous efforts trying to develop the vaccine for HIV, and all of that failed. But we have really good and working antiviretics from multiple different mechanisms that can be combined and basically put that disease under the roof, and that person can, with HIV, can live for decades these days without any problems. So it really depends on how you want to tackle and how you can find the way how to basically go to against that specific disease. One typical problem, especially in cancers, is that something is overproducing itself. And in that case, you want to inhibit it. So that's quite a lot of CDK and ALS kinase inhibitors and so on, where you basically have a kinase and you want to inhibit it in order to stop overproduction of the growth of the cancer cell. Another thing is that you have some kind of a receptor, and that receptor is firing by itself, and it's basically giving a lot of noise towards the cell and puts a lot of stress on it. In that case, it's similar because you basically put there an antagonist on that receptor to slow down the response, and that's how painkillers work. Because that's the COX-1 or COX-2 receptor on our cell. It's firing to us, giving us the information that something is paining us. And you want to basically put this signal out, and therefore you put there an antagonist for that. So that's quite similar to the inhibition. What is more and harder to develop is the agonist, where you want to switch inactive receptor to the active one, basically to activate it. There are some ways how to do that. For instance, if you have the structure crystallized with agonist, then or the structure of the active transporter or active basically conformation of that specific protein, then you can develop specifically for that. And by that, you basically can develop some agonists. It's not always the case that you have the agonistic structure ready in the database, because with the inhibitor, it's much easier to develop it. Sometimes it's a regulation peptide. And in that case, it's really hard, because if the protein or if the peptide does not usually have some defined structure, so it's really hard to define something that would bind it as a small molecule. And in that case, you basically have to switch towards the biologicals, because the peptides is something that our immunity antibodies are done for, because they look for the peptides and attack them. So with that, antibodies, this is the perfect way for them to be used. Yeah. And also with RNA, there was one drug which was in RNA last year approved. And it's not much yet, but it could be that you can do some silencing or some aptamers drugs and so on. So usually we deal with small ligand with proteins, and that's taken by the Emil Fischer, who developed his lock and key analog of the silver bullet as a drug, as a silver bullet, which was pointing towards the protein as a target. We can use that analogy, but as I've already shown you in the FDA-approved drugs, we are using that for approximately 50% of the drugs currently. For the rest, we are usually looking for the antibodies. So what are the drug targets which we usually take? Typically, we are looking for the compounds, for the protein targets that are somehow connected towards the regulation of the cell. And there are multiple ways how to do that. And the first one is G protein coupled receptors, GPCRs. And those ones are on the plasma membrane, waiting for some hormone going from outside. And basically we can use that in order to basically use that road to basically block or to either antagonize or agonize them in order to put their signal off or on, depending on what we want to achieve. And that's 1/3 of our drug development. The next group is ion channels. Ion channels, because they basically, again, this is the regulation about how the drug, about how the cell functions. And as well, similarly, the nuclear receptors are regulating how the DNA is read. So this regulation of how the DNA is read is also quite important for what proteins will the cell afterwards be generating or not generating. And interestingly enough, quite a lot of development, especially in small molecules, was done on kinases. But as you can see, this is what was targeted, and this amount of the kinases was really large, 10% of the drug development, but only 3% of them actually final drugs. So this is one of the groups which unfortunately does not work well. And one of the major reasons why those kinases did not work out was that all kinases are kind of similar to some extent. And therefore, if you are developing something which is killing kinase, then quite often it's killing another kinase as well, which can be going against what you want to achieve. So it's much more useful to go towards the signaling parts, because those are the ones which actually have some impact. So if we use the ATC code, which is basically the way how the doctors are basically going where the drug is going for, we can say that we have covered all of those diseases somehow with drugs already, but some parts are missing, and some parts, especially immunosuppressant and antineoplastics, are seeing quite a lot of new effort and new registrations. Unfortunately, this is from the 2017, so I haven't found an update on this study. But it's quite interesting how the development of individual groups basically was changing along the years. And well, it's good to mention that actually 60% of the drugs targets are membrane bound. So either it's a membrane receptor, or it's enzyme which is found to the membrane, or it's ion channel, or it's transporter, or there is 0.5% of the cell membrane itself. Yeah. So there is quite a lot of effort going on in different groups, and the development is quite different in the individual years. And I'm going to the last part of my lecture, and that's the comparison between small molecules and biologics. There is the one way how to look at it is the size and complexity of the antibody in comparison to the aspirin. Of course, most of the drugs which are now currently developed as a small molecule drugs are above 500 Daltons. So it's 5 times bigger than the aspirin. But still, 5 times bigger than this is still much, much smaller than the antibody. And the problem with the large drugs is that in order to generate the large drug, you have to purify it from some kind of a genetic code, and you are putting things into the living organism in order to produce the things in bioreactor and then to purify it. And the problem with the biological cells, let's say, is that they tend to live, and as living is a constant change, what was working a year before might not be working a year after. And for instance, I was collaborating with the Teva Pharma company in Opava, which is on the north from the city, and they are making the immunosuppressants from a specific fungi, which is living on a weed. And the problem is that they really have a problem to find a weed living around the area that they could put the fungi on, because all the weed around is basically resistant to that fungi currently. So they basically have the fields elsewhere, and moving the wheat, which is with this fungi from quite large distances because of this resistance to that, and they are trying to overcome it somehow by some kind of bioreactors where the fungi would be produced. But it's not really easy to generate it, and since the fungi is making the starting product, they have to start to look around to find another source of the starting compound, or to start from something else. While in case of chemistry, if you are an organic chemist as I was at the beginning of my career, usually when you follow the recipe, then you get the drug as it should be. Yeah. As I said, that there is one reason why I'm now a theoretical chemist. For some reason. Well, if you have small molecules and we want to do the drug design, you have plenty of options. You can do the structural-based drug design, for instance, docking. You can do the de novo design. You can do with the ligands itself. You can do the similarity search, pharmacophores, QSARs. There is plenty of things you can do with the small molecules. And even if you don't know the ligand and you don't know the target structure, you can still pre-select the compounds to test for the toxicity, for instance. But with biologicals, these are all biologicals. It can be blood derivatives, blood components. It can be individual proteins. It can be human tissues or xenotransplantation. It can be cellular or gene therapies. It can be vaccine, allergic extracts. Quite a lot of things that can be used, and each of them follows its own part. And for instance, we still don't have a way how to make the artificial blood, unless you are Dr. Morpheus. He had it. So, and another problem with the proteins is that usually when the protein is produced within the cell, it don't stay as a pure protein by itself. It's usually post-translational modified by some carbohydrates, phosphorylated, glycosylated, lipid attachment, cleaved. There's plenty of things that the cell is actually doing towards the protein. And therefore, it's necessary to do those steps if you want to produce that specific type of protein, for instance. And that's slowing the progress there. But these days, there is a protein engineering toolbox, which just recently, like one month ago, got quite boosted by the protein large language models. Similarly, as OpenAI boosted the chatbots with its ChatGPT. And now it's possible to develop the protein designs for specific structure, a specific purpose, much more effectively than it was previously. So just a take-home message, these protein drugs are important. We will not be speaking about them much, but we will be in the few lectures later. And for small molecules, it's really important to find out what you are actually developing and to tune your approach towards what the drug should be doing. And that's it. So, do you have any questions?
Maybe I will have a question. When you talked about those similarities of new compounds,
just wait. I will just.
No, because this idea, what I first come to my mind, those compounds which are similar to the previous ones, they can be repurposed compounds. So,
Yes.
so they shouldn't act on the same target or the same mechanism. And what is the proportion of such compounds among those highly similar ones?
Usually those highly similar ones are going via the same mechanism.
It's not necessary.
It's not necessary, but usually they do.
Well, they didn't such analysis.
No, they were looking at the financial part of the drug development. So not towards the individual mechanisms it's per se.
So regarding these deuterated drugs, so do you think it will change soon the regulations or the patent makers to include always deuterated form?
Well, I guess that it will happen soon because this is actually this deuterated one seems to be on a spotlight, and yeah, I guess so. But the problem with that is that if you put there the deuteration, the deuterium instead of the hydrogen, of course, when the drug is large enough, it won't have any much difference. And if you put it into a clear water, you will have a hydrogen deuterium exchange as well. But how you can basically say that at the beginning where you actually have the deuteriums in order to do the proper registration via patent? That's tricky, I would say. How to cover it? Yeah.
Do you think there is a reason why the cultural drugs are more or less approved than healthy ones?
Oh, you mean like the new drug types and so on? Because the major reason I don't know, but I guess that the reason why is that so? I guess is that you are trying a new mechanism of action, and as such, that's the reason why it's failing, I guess, because there is plenty of studies where the pharma companies were taking the papers from the good journals about the new mechanisms in disease, basically cancer disease, and they've tested those studies and they were not able to reproduce them. So I think that since you are looking for a specific new mechanism with those new compounds, then it can happen that this mechanism is actually not the one that's actually the important one. I have one example of that, and that's camostat. Camostat is a drug which is TMPRSS2 inhibitor, and TMPRSS2 is one of two proteins on the human cell that's actually interacting with the SARS-CoV-2 particle, because the spike is interacting with the AC2 protein, but then it's cleaved by TMPRSS2. So the reasoning about testing this drug was to use the inhibitor for this TMPRSS2. That should basically be blocking the way how the SARS-CoV-2 would be going inside the cell. But there was another mechanism how the SARS-CoV-2 is able to go into the cell, which is not using this TMPRSS2. So sometimes, even if you have a right mechanism, there is way around how the disease can proceed. So sometimes you are just unlucky. Yeah. So now I need to set up Zoom here again. Yeah, the problem is that it was closed down by someone. Have you done it? Yeah, because there is no Zoom again on this computer. So I need. Oh, it is. Yeah. Yeah. Jo, vypni.
Můžeme, že ne? Ale musíme k tomu být protože jsou prezentační módy, jsou to dobře. Melissa, James, can you hear us?
Yeah, can hear you fine.
Yes, I can hear you.
Melissa, James, can you hear us? Can you start?
Yeah. Yes, I can hear you. Can you hear me coming through there?
Okay.
Unfortunately, I still can't hear you, James.
Testing, testing. 1, 2, 3. Right, how about now? Yeah, it appears that your speakers are not connected because Barbara is on the call and can hear me. I'm just going to keep talking until you hear something coming out of the speakers. Hello, we're from ChEMBL. Can everybody else on the call?
We are hearing you, but not much. Yes.
But people in the hall can't hear. Yeah, you're getting something because you're getting reverberation off that teacher account now. I'm hearing what you're saying.
Dokončíme, to nejde.
Musíme to spíš tady. A tady no.
Try to speak.
I think you might need to turn off the mic on one of those computers.
Seems like now you can hear us because now you've heard you well.
Yeah, I can hear you. I'm only hearing one of you. I'm not hearing echoes.
Okay, cool. So now it's working. Can you share your screen?
I don't know.
Share the presentation.
Microsoft PowerPoint. Right, should be able to see my presentation window there.
Yes, yes, we see everything now. Yeah.
Okay, brilliant. Hello, I'm James and this is Melissa. We're from the ChEMBL group at EBI. We're going to be doing 2 back-to-back presentations on ChEMBL here. I'm going to be explaining what ChEMBL is, a high-level overview of how to use it and what data's in it. And Melissa is going to be doing a live tutorial and walkthrough of ChEMBL. If you've got any questions, please put them in the chat while we go along. The way we're going to do this is Melissa is going to try and answer any simple queries you've got while I'm doing my half, and I'm going to be doing the same for Melissa, because that way we don't need to interrupt the presentation to answer questions. If you've got anything that's a more involved question, if you can leave it to the end, we're going to have a 10-minute Q&A session, and that'll be best. But sort of simple clarifications and questions about the slides you can put in the chat. Right. So we're going to have 25 minutes of introduction to ChEMBL from myself, and then 25 minutes of tutorial from Melissa. And I'm going to be going through with you what is ChEMBL and how is it structured, what data is in there, how we extract and curate the data from scientific articles and depositions from chemists, how the drug data are curated and annotated, and a little bit about how you can use the graphical interface and the API. And then Melissa will go through the nuts and bolts of how that actually works. So what's included in ChEMBL? ChEMBL is an open access data warehouse for drug discovery. It's manually curated, it's high quality, and it's fair. So it's findable, accessible, interoperable, and reusable. And the manual curation lets us make sure that data is findable in similar ways. We'll go into that in a bit more detail later. The core of the database is primary medicinal chemistry literature and donated data sets. We've got a deal with a company that passes through major journals in the field and brings those literature papers in. We also accept data sets from other people's papers. And we look at all research starting from about 1980 that fits our criteria. So you can see we've got industry data sets, we've got collaboration data sets, open access public sources, and anything to do with open clinical data and licensing. So when ChEMBL started, we were mostly a publications database. And since then, we've become much more a datasets database. You can see the growth from version 9 to version 31, which is the version before last. And over time, we've increased the number of deposited datasets we've got, and that's grown at a much higher rate than publications. And of those publications, there's a bit of a long tail there. Most of the publications are from sort of the first 4 journals, and then the rest of the journals make up the rest of it. So it's kind of a few core sources and a lot of ancillary ones. So summary, this is for ChEMBL 32. You're getting a sneak peek of what we're bringing out in mid-February. And that's going to have 213 deposited datasets, about 86,000 publications, 15,000 drug targets, 2 million compounds, and 20 million or so activities. You can search the database by any of these categories. So we collect a number of statistics for the compounds that go into ChEMBL. So we look at the molecular properties. We've got 2-dimensional structures. You can search by that. You can draw it. You can upload a structure as a search pattern. We've got calculated molecular properties like the log P, the molecular weight, Lipinski rule of 5 parameters, etc. We've got protein target interactions. We've got cell and organism level assays, so ADMET data and the like. We've got bioactivity data from any deposited assays or any literature results that show that. And we've got a curated set of drugs and clinical compounds, and those are annotated with known therapeutic targets and therapeutic indications. So if you look at the website and search for a compound name, you'll see a set of tabs at the top, and those are the main entities that we look at. So we look at compounds, and we've got chemical structure and properties from experiments using that compound. The targets, which are the biological system where the measurement was done. So that's going to be the lowest level target. So rather than human, if it's a human protein, it will be telling you the protein. And that's normally the protein, cell, or whole organism level. There are assays, which are a description of a specific experiment. Important thing to note of ChEMBL is if the same assay protocol is used in 2 different labs at 2 different times, they are 2 different assays because it's not the protocol, it's the instance of doing that thing. We've got the documents where the data are reported, which will typically be either a scientific paper or a deposited data set with name and information on how to find that. And then we have cells and tissues in which the bioactivity data was measured if the experiment had information for that. So we use a service to extract literature data from papers, and from a given paper, they will attempt to get us the reference, the assay that was used, the target of the compound, the organism that the assay was done in, and structural and property information on the compound itself. So this starts with manual extraction. They're extracted from the publication's full text. It's comprehensive. We get them to extract all the quantitative bioactivity measures they can, and we load all those into the activities table. And we take multiple data types. So anything on that list, we will be loading into ChEMBL if that's in the paper. So binding, functional, efficacy, pharmacokinetic, toxicology. We have both manual and automatic curation steps. So we have a standardizer, and you can see the reference below. We'll be sending out a handout with links to our code base and publications on it after this. And the standardizer automates error checking, compound standardization, and salt stripping. So we know that any compound structures that are being included in ChEMBL are both free of errors and drawn in a consistent manner. We calculate additional data and properties using this pipeline, as you can see on the right. And then if we have any remaining issues, ChEMBL curators correct those. So those issues will be revealed in loading. So it might be invalid structures, name structure mismatches, or stereochemistry errors. I report those along to the curators and they fix those. So for activities, we standardize the data where possible. Because a large proportion of the data is just dose response curves against protein targets, we're looking at, say, IC50, we use something called a p-ChEMBL value, and that's calculated when the relation is equality and units are standardized to nanomolar, and it means that you can compare different data types using a sort of general magnitudes number. You can find more in our ChEMBL documentation. So the assay type, they're always written down for assays, and we only have 6 categories that they can fall into. And those are functional data, so biological effect, Adasme data, so pharmacokinetic data like T1/2 or bioavailability, etc. Toxicity data, molecular binding, physicochemical properties, or if we can't assign it to one of the above categories, we'll put it into unassigned because we are using a limited vocabulary, so you can actually sort on a small number of defined categories there. So for the curation steps, we standardize the activity types. So because papers might have written IC50 in multiple different ways, it's always going to be written as IC50 in that way for us. If there's logged values or negative log values, we anti-log them and we get them back to the observation, not the log of it. We then standardize values and units. So if it's a molar concentration, it will always be recorded in ChEMBL as nanomolar so that you can compare them directly without having to do the maths in your head of which ones are millimolar, which ones are micromolar. You can just look down the column and the larger number will definitely be larger. We flag potentially incorrect data, so ones that might be out of range, ones that might be non-standard units. It affects data that might have a very, very small amount of effect and not be submitted as negative. And then we calculate p-ChEMBL values. So for any of these particular measurements, IC50, XC50, EC50, etc., we take the anti-log 10 of it, and that will be a p-ChEMBL value. So between all of these, you can get a rough estimate of the size of the effect. And then we flag potential duplicates where information might be about the same compound, target, type, or value. And finally, the curators do a manual step to fix the erroneous data, either structurally or otherwise, that we get. So if we believe that data might be out of range or non-standard, that could require me going back to a depositor and asking them, did you mean this? Can you resubmit? If it's a structural one, then that's something we can often fix in-house. And this gives you some idea of how we curate the assays by type. So is it molecular? Is it non-molecular? And then we can split that into our could not find anything else categories: tissues, cells, organisms, or molecular targets of nucleic acids or proteins. I've got a bit more information for you on the right there. So we could see that assays lacking biological material will go into the null category, whereas on the other end of things, where it's a very specific protein-based assay, it's going to go in protein category. So what method do we use on those curation tasks? We get the target types and assay types from whatever the depositor describes the assay as. And we standardize the names for tissues, cell lines, etc., by using controlled vocabularies. So Uberon, cell source, and so on. And the assay formats are mapped to the BAO ontology. So again, where possible, we're using controlled vocabularies, so you can search on those terms and return all the relevant assays from ChEMBL. And common manual curation tasks are curation of the undefined type, remapping of toxicity assays from activity to toxicity, and remapping of GPCR assays that measure secondary measurements from binding to functional. So what target types do we have in ChEMBL? We've got a little tree of that there. So examples of non-molecular targets would be the mitochondrion, a whole organism, or neuronal cells. Molecular targets would be nucleic acids, protein nucleic acid complex, or small molecule, or indeed protein. And under protein, we've got subcategories of single protein, complex, complex group, families, or protein-protein interactions. And proteins are sort of disproportionately represented here. In 31, we've got 8 million bioactivities on protein targets and 12 million bioactivities on all non-protein targets. So when we're looking for drugs and clinical candidates, we've got a variety of sources that we're going to be pulling data from. So clinicaltrials.gov, British National Formulary, withdrawn drugs, pharma pipelines. And for all of those, we annotate by disease, we name and structure map the compounds involved, we annotate the mechanism, and we annotate the drug properties. And that little graphic is the ChEMBL dashboard for what known drug properties are available for that particular compound. And these data go into both ChEMBL and also into Open Targets, which is another project that involves EBI as part of the consortium and is more target focused, whereas ChEMBL is more based on the compounds themselves. So that little dashboard that we've got there for a drug, I've got an example here for celecoxib, and the top row is molecule-based properties. The bottom row is product-based properties for approved drugs. So on molecule level, we've got what type of molecule it is. We've got if it's rule of 5 compliant. Was it the first drug in its class? Is it chiral, racemaic, or achiral? And is it a prodrug? And relating to its use as a drug itself, we've got method of administration, so oral, parenteral, or topical. We've got if it's got a black box warning or has been withdrawn as a drug. And we've got the availability type. So if those are coloured, we have information for that. So we have link outs from ChEMBL to other molecule-based databases, and we use a service called Unichem, which I also actually work on for that. So Unichem is basically a chemical directory. Data from ChEMBL, PDB, ChEBI, and other internal EBI services can be mapped between each other and external services. So if you have an identifier for a chemical on any of these databases, you can find if it exists in any of those other databases. And it's got about 170 million compounds, 40 different sources. But what's important in ChEMBL is Once you've got your structure, you can look either at the output programmatically from one of our web services here, or in the tabulated section of the website, where you'll see cross-reference for the compound in question in all these external databases, and you can go and get a broader look at that compound. How do you get access to the ChEMBL data? Well, you can go through the website, which has a graphical interface. You can do web services where you're making your query through a web address that then returns some results. There are direct downloads of the database available, and there's also access through the semantic web relational formats. But of these, which should I use? Well, you should use the API if you need a repeatable query, or you want to embed that query in a script that you're running a sort of large batch job. If you don't want to download the full database to do that, because the database is quite large and you need to set it up, or if you want easy permanent access to the data via a URL web link. So if you want to share that query with someone else, you can share them the API URL and they'll be able to pull the data. The web interface is great for browsing, especially if you want to go deep into a topic. So start at a compound and work down through targets and assays, because it's got a lot of exploratory tools built in there. And if you need to do a lot of heavy work on the database, so running multiple queries with a lot of input data, or you have complex bespoke queries across a lot of database tables, and you're happy with SQL, then I'd download the database and do that. It's also the only way we have for you to access data from a previous release. The website and API will be for the current release only. So we only provide the most up-to-date data directly on the website. So a web interface is we've got a landing page with a search box at the top, and you can search by any text string you want to look for in any record. It could be a disease name, could be the name of a author on a publication. You could search by chemical structure directly in there, chemical identifiers or protein identifiers from other databases. Or we have an option for a blast search, so you can put in a nucleic acid or protein structure and search for matches on that. This then takes you to a browser page where you can explore the data sets, see distributions of them between, saying here we've got whether the structure results are small molecules, something else. You can apply filters on that data, and you can see relationships between the entities that you've returned. And for individual entities, you've got a report card where you can see the details of one item. I'm going to zoom through these quite quickly because Melissa will be showing you them. But a free text search, if you put in a term up top, then you will then get all the results of that across all the things in ChEMBL. And you can view different presentations using menu there, so table, a graph, or a heat map, and investigate the text of your choice. And then you can see specific report cards in the window at the bottom. You can upload or draw a structure using a Smiles string, for example, or directly drawing it in Marvin Draw here. And then that will give you structure results, and you can choose if you want to see highlights of a structure. You can choose if you want to see similarity maps to similar compounds in ChEMBL. You can also search by ChEMBL IDs. So if you know the ChEMBL ID for a molecule or an assay, you can paste them into the search box as a list and return all those results. That will take you a while to get back because it's run by a job on our servers, but we'll give you details of how that is going. As usual, you can go to the dashboard, you can explore the results by compound, assay, etc. And it will also give you a summary table of everything that was found. For Blast Search, you can enter a sequence in any format that's supported by the EBI Blast service, and you can modify the parameters for, in this case, the Blast P search with the little menu below there. And in this case, again, we'll give you a progress meter and then results. And those results will give you the Blast E value and other scores, and then it will link to the ChEMBL records for the Blast hits. And right on the front, we've got the browse page where you can click on any particular entity, so compounds, assays, targets, and just go straight to absolutely everything in there and start filtering by whatever types or restrictions you're interested in looking at. Report cards for each individual chemical here. You've got the structure, you've got a summary table of information, and you can jump to various sections of the page. You can also do a structure search based on the compound on that report card directly from that page. I'm going to briefly cover APIs before Melissa takes over. If you're not familiar with what an API is, it is a web address that lets you ask a website a question. So rather than browsing to a specific web page, if you put in something to the API address, then it will ask, in this case, for a molecule where the preferred name in ChEMBL contains acetaminophen. And by tapping that in, we get the question. And the question has the stem, which is where the API lives. It has the resource, which is what type of category you're asking the question of. It's got a field that you're filtering on, and then it's got the filter that you're using within this field. And important things to know, use of underscores and formatting. But again, Melissa will cover these things. And we can return results of these by default as XML, but also other machine readable formats like JSON and YAML. So if you're building this into a script, it will pull back something that is very simple for your script to read back in. So just to give you an idea, that's what it looks like on the web interface if you're looking at acetaminophen. And that's what is pulled back from the API programmatically. You can see one is quite easy for a person to read, and another is quite easy for some computer code to parse. Best practice for ChEMBL is use a web interface to have a look around. Then once you've mastered that and understand what we've got, look at the API. To get started on the API, you can look at some example URLs. And again, this will be on the handout. We'll be sending the examples, but you can use to do some dummy searches and get to grips. Check the data output. Does it give you everything you need? Are the activity data for both the parent and salt drug forms present if that's what you're looking for? Or am I happy just getting the parent form? We've got a full schema for ChEMBL available if you're familiar with SQL schemas of this dress. And we've got some interactive API documentation. These links will all be on the handout, so I'm going to zoom past those to get to the actual workshop. And I'd just like to thank other members of the ChEMBL group, including Barbara, who is listening in on this call because it's our first time presenting, and who has contributed some of these slides. Fiona, who also contributed some of the slides to the initial training materials, and David, who did that lovely breakdown of the web interface that I ran through with all the red arrows on. I'm going to hand over to Melissa now. Thanks.
Yeah, okay, so I will share my screens now. I hope you see it. So do you see my screens?
I can see your screen, Melissa.
Okay, that's great. Then, yeah, my name is Melissa Adasme. I'm a data mining scientist at the ChEMBL team, and I'm gonna guide you through this tutorial that is a bit more practical than the background that James just did. So today, we're gonna cover a hands-on exercise on a real example, and we're gonna use ChEMBL to retrieve relevant data for that example. So we're going to try to find available inhibitors for a given target. We're going to characterize such inhibitors, and we're going to retrieve the activity available in ChEMBL for them. Finally, I'm going to show you how to cross-reference the ChEMBL data to other sources that might be relevant. And if we manage the time also, I'm going to show you some examples of using ChEMBL API with our own example, and finding ChEMBL data for a target or ligand of interest. So first of all, if you have a laptop with internet access, which I'm assuming, please try to go to the ChEMBL website, which is the URL I'm showing here in the slides. If not, you can also Google it. It's very easy to find. And yeah, so a bit of background of the example we're gonna work on today. I'm pretty sure many of you have heard or are at least a bit familiar with the AVL kinase inhibitors. And this is mainly because the BCR-ABL fusion mutation is very common in patients with different types of cancer. Actually, there are several drugs that have been approved to treat cancer by targeting BCR or ABL. The most famous one is imatinib with some derivatives such as nilotinib, dasatinib, bosutinib, and most recently one ponatinib. But the key part here in this story is that ABL has a type of mutation which is known as the gatekeeper, which is from threonine 315 to isoleucine, and it's causing drug resistance to these drugs that I show you here. So what we're going to do today is we're going to use ChEMBL to extract data for these drugs for BCR and ABL targets, and we're going to try to see what we find out with the activities that we see in ChEMBL. And as a first exercise, we're going to try to find approved ABL kinase inhibitors in ChEMBL. And for that, we need to do 3 main steps, which are the ones that I show you here in the slides. I just leave the screenshots as a backup for you to come back later. But I'm going to try to do it live with you, so try to follow me. If you get lost, just write it in the chat and James will try to help you catch up. So this is the main web page of ChEMBL. And as James already explained, we have the browse bubbles here on the left, which are really nice. And in each of the bubbles, you see each of the resources with the current number available in ChEMBL. So since for ABL, we know that it's a mechanism of action that we're familiar with, we're going to start our search using the mechanism bubble. So just you can click there in the bubble, and you will see a list of all the records available. So all different mechanisms that exist right now in ChEMBL. So 6,300 records. But we want the mechanism for ABL. So what we're gonna do is on the right side, in the search field, we're gonna type ABL, but be careful to first, before remove the wildcard that is there by default, because that will allow the search to be a bit more permissive, and we want it to be as strict as possible. So we're gonna type ABL, click on the search button, and you will see that we find 34 records containing the ABL keyword. Now, just be aware that this is a keyword search, so there might be things that are not exactly what we are looking for, but for some reason they contain the word ABL and were displayed, but we're gonna filter this later. Now, the next step is that we want to find only approved drugs. And for that, we can use the filter section on the left, which allows you to filter by several different types of, for instance, organism taxonomy, target organisms, protein classification, or action type, among many others. But the one that we want to use right now is the patent molecule max phase. And we're going to select the max phase equal to 4, which is the approved drugs. So if you click there, you will see that now our records were reduced to 17, which are the approved drugs for the ABL keyword mechanisms. Now, this is the main core of the results that we have found so far. And using that information, we're going to try to answer these questions. The first one is how many approved drug targets ABL or BCR-ABL? And for that, I provide you already with the ChEMBL ID. Now, we're going to try to do this very quickly. And since we already know the target ID, you can sort by the ChEMBL ID. And you now easily will find the IDs that you are looking for the specific targets. So in this case, we have nilotinib, regorafenib, dasatinib, bosutinib, imatinib, and as you can see here, there are some IDs that are not exactly what we're looking for. So you just ignore those because this is not the ABL or BCL. And in the end, what we will get is only 6 drugs targeting those ABL or BCR-ABL. What year was imatinib approved? 2001, that you can easily see here in the first approval column from the result list. So if we search by imatinib compound, you can search by the year, and you can see the years of approval for all the other drugs. Then, what are the drug indications? I already put the results or the answers here. But for that, what you can do, you can go to imatinib compound in the list and click in the ChEMBL ID. That will give you the compound report card that James explained before in the slides. Now here we have a summary of all information available for that specific compound in ChEMBL. And on the right, you have the content with all the different type of information. So we are interested on the drug indications. If you click there, you will see there is a list of all indications associated to this compound, but you need to pay attention to the max phase for indication because we just want the approved indications. So you can click on the arrows and you sort them by the max phase indication, and we have only 2 records with approved state, and that's neoplasm and leukemia. And that's what I wrote as an answer here. And in the final question, what other proteins are involved in the mechanism of action of imatinib? You can again go to the compound report card. On the right, there is another section called drug mechanisms. And here you will find all mechanisms associated to that compound. Now, we already know that BCR-ABL fusion protein and tyrosine protein kinase ABL are associated to the compound, but the other 2 are the ones that we are looking for. And this is how you answer all the 4 questions, just using the main records and the main report card of the compounds. Now, in the second part, it's a bit more tricky, but we're gonna try to retrieve activity data for the ABL inhibitors that we just identified. So what we're gonna do is go back to the main list of the 17 records associated to ABL with approved drugs. And on the top of the web page, you will see that there is the option for browse drugs. If you click on the browse drugs, you will be directed to the different display of all the drugs that were associated with that mechanism. And as James already mentioned before, there are different ways how to show this type of data. It could be the cards, the table, the graph, or the heat map. So we are gonna select the heat map. And we are gonna try to see all this data in a most comprehensive way. Now, what you see here on the heat map is that all the compounds are associated to that mechanism are listed on the left and the targets for those compounds on the top. Now, what we can do here is to search for each of those pairs, for instance, the first compound, imatinib, binding to the ABL tyrosine protein kinase, you can see all the activities. And if you click and see all the activities, you will have a list of all the reported activities for that specific compound with that specific target. So I want you to have a look to that data and try to answer the following questions. What is the binding constant for imatinib against ABL with the wild type protein, meaning that it does not have any mutation associated, but also to the mutated version with the threonine 315 isoleucine. And I give you some hints, like to use the filter section to get the dissociation constant. The filters, remember, are on the left side, to check the side description to see if it's the wild type or a mutated version. And remember that several activities can be listed for a specific compound and the same target. So usually what you do is that you take all the activities and just provide a range or the mean, the average. So try to get all the values and then repeat the same, but for the mutant type of the protein. Now I'll give you just a couple of minutes so you can process the data, work on your own, try to see what you find, and then we can discuss all together the results. some tips that I can share with you is that on the top of the web page, you can show or hide columns of interest. So if you click there, you will see which columns are being displayed right now and which ones are not. One column that might be very helpful to detecting variant mutation is the number 41. So if you click there, you should now be able to see in the last column the assay variant mutation. And it's telling you immediately if there is a mutation, which mutation is, and if there is no mutation, you can double check on the assay description if it's really the wild type or it has another variation. And we have several activities reported for this compound. So just try to filter out exactly what you need. And pay attention to all the types of activity and the details like the standard value, if it's standard unit, nanomolar, micromolar, if it's IC50 in this case. But as I told you before, it's better to filter out by the dissociation constant on the left. So if you click there, it will be restricted to just dissociation constants. Okay, so I hope most of you already kind of got the idea how the activities are reported in ChEMBL, how to filter them. If not, we can discuss this further. But maybe let's start checking the results. As I told you, the activities are not exact value. It could be different reports from different groups testing the same compound against the same target, so they might vary. But usually trying to get the range or a mean, something that is representative for the whole set, it's very useful. So for the first question, what is the binding constant for imatinib against the ABL wild type protein? We get a range between the 1 nanomolar and the 106 nanomolar. And this is, of course, as I told you, depending on the assay publication. So it ranges in the nanomolar, the activity. What is the binding constant for the imatinib against the mutant variation? You can see that from the different assays that I also put the IDs here so you can double check later. It's from 6.2 to 10 micromolar. So according to the definition of the activities and the dissociation constant, we can see that imatinib for the mutated version of the ABL is not as effective as for the wild type. And this is what I was explaining you in the background. It seems that it is effective for the wild type, but the moment we see the mutation of the threonine 315 isoleucine, the activity dropped down and is not effective as it should be. Now, when you repeat the exercise for the dasatinib and ponatinib drugs, and this is a bit tricky because we don't have dissociation constant for both compounds in wild type and in mutant, but if you use all the measurements such as IC50 or Ki or whatever is available, you can still make a comparison of the action activity. And you can see that the dasatinib behaves in a similar manner as imatinib. It's very active against the wild type, whereas not as active for the mutant type. But for ponatinib, it's not the case. Ponatinib is equally active or ranges in the nanomolar for both cases. So what we can conclude from these results extracted from the ChEMBL web service is that imatinib and dasatinib bind with at least 25 to 60-fold lower affinity to the mutant type, but ponatinib is equally potent. And if you want to explore this further, in ChEMBL, we provide the cross-referencing part. And this is also something that James already mentioned. So in the compound report card, we have on the right part, the section which is called Unichem cross-reference. And if you go to that cross-references, we provide already the mapping to all other sources that might be relevant for your studies after you got the data from ChEMBL. In this case, if it's the imatinib drug or the ponatinib, you could get the 3-letter code and go to PDB to do a bit of search and extract all the structures available for that specific compound with that specific target, and maybe also explore the ligands environments to see what exactly is going on there with the mutation, if the mutation is involved in the binding site, if it's affecting the binding of the drug, or why it's interfering with the activity. Now, of course, this is not a training for PDB or structural data, but just to show you that the options are there, and that if you do that, maybe you can try it on your own later, you will see that there is something that we can say to explain this. And it's because imatinib and dasatinib, they both have H bonds with the threonine in question, whereas ponatinib, it only has pi cation interaction. And it seems that when the mutation happens from threonine to isoleucine, ponatinib can deal with that and can accommodate easily in the pocket without any effect or negative effect in the binding. Whereas imatinib and dasatinib lose that strong binding and they are affected. So this is how you, starting from ChEMBL, can construct a whole story explaining and using the data that we have already available. Now, the same as we did right now with the website, you can do it with the API. Now that you are more familiar with the mechanism of action and that you can identify the targets by their IDs or the compounds of interest, you can maybe try with the API. And I highly recommend reading the API documentation, because here we provide everything, even some examples, how to start working on, the resource available. We have activities, assays, binding sites, drugs, compounds, exactly what you see on the web service, you see it here. And also the supported output files, or the ways how to use the filters that James was previously mentioning to you. So for instance, for our case, we can start just searching all mechanism of actions by defining the mechanism resource, and you will get all that is available right now in ChEMBL. But if you want to be more restrictive to our case study, you can provide the targets IDs and filter for those specific targets. And you can provide also maybe say, I just want approved drugs, so max phase has to be equal to 4. Or maybe you want to be a bit more permissive and you say, okay, max phase could be 4, but also could be clinical trials 3, maybe, as you want. And that's the good thing about the API, that you can be the owner of your queries and you can play them as you prefer. And of course, we also provide different formatting, not just XML, so you can also work with JSON files or the best that suits you to the things that you're doing. And since we don't have much time, I just want to share with you 2 bonus exercises with some questions, answers, some hints, how to do it, what to do, what to avoid. And the first one is to find ChEMBL data for a protein of interest. You can use the UniProt IDs, you can use the sequence, and I give you some questions with answers so you can play with the data on your own. You can test, and some things to do and not to do, as I said before. And also how to find ChEMBL data for a ligand of interest. You can search by the name, by the structure, or any other identifier that you can provide, and we map it. So with that, I would like to finish. And if you have any questions, just, I mean, now it's a moment. We have, I think, 10 minutes left or more. So whenever you want to ask, now is the moment to do it. We are happy to answer. So thank you.
Thank you. So do you have any questions? Anyone inside? Yeah.
One technical question. How often is the database update?
Have you heard?
No, sorry, I think we cannot hear you well.
Oh.
The question was, how often is database updated?
So right now, I think we are updating it every 6 months. The goal has always been like to move to every 3 months, but right now officially is 6 months. Every 6 months, there is a new release.
Hi, my question is, you said about the sources is also pharma pipelines. So how come the pharma companies want to share their pipelines?
I'm not sure they all share the pipelines by itself. I mean, we have projects working on that going on. If there is collaboration, of course, there is always an agreement beforehand, how much will they share or not. I think it's usually about data sharing more than pipelines itself. But I don't know, James or Barbara, you can both correct me if I'm wrong.
Yeah, so we get depositor data from anyone who chooses to submit it. And if we get something that's from a collaboration or any sort of pre-competitive data sharing, then that's the kind of thing that might go in. We don't sort of go out and solicit for commercial information, but when consortia or individual labs are willing to share it, we'll just take those submissions.
Thank you. There is a question in chat: how to access ChEMBL data ChEMBL data through Python notebooks?
Let me go and dig that out, because we do have a Python library for ChEMBL. You can use the API and just query that with something like request library. That's what I tend to use in my own pipelines.
Question regarding the standardization procedure. When you register salts and with some biological activity, is this data automatically attributed to a parent compound or not?
I'm not sure I got the question. It hears very far away, sorry.
Okay, the question was whether the salts, the data on salts, whether they are uploaded towards the parent compound or how they are dealt with. Is it?
You mean if any activity appears and we already have the compound, we have an internal mapping that usually makes like sure that the correct compound is assigned, but also we have manual curators that process the data and make sure that the right mapping is occurring internally. So this is how we usually link the new data. Every time that a new resource is released, there is a new processing of the data that comes from the literature, and there is a manual curation that takes care of that.
Similar to that, do you somehow deal with the prodrugs?
With the prodrugs, I'm not sure.
If it's possible to basically look for a prodrug and the drug pair, to gather how many prodrugs and what type of prodrugs are used?
I think it should be possible. I'm not completely sure. James, I don't know if you want to add something, because to my knowledge, I think we do not, or so far we do include prodrugs, but not the search exactly. So James, do you know?
It is annotated on the report card page. It's not a filter I've used, so I can't confirm it exists. Best thing is I'm just sending the handout here, and we can send that to the organisers to mail around after this. But we've got full documentation on the web service and on the interface. So if you've got a query you wonder if you can do, or a field you want to check how to filter on, then that's the best place to start looking. Because, for example, although I load the data to ChEMBL, I don't make extensive use of the web interface or API, so I can't, off the top of my head, answer that sort of question.
Thank you. Any other questions? Okay. If there is no other question, thank you for the nice introduction and tutorial about how to use ChEMBL, and also for the documentation, which I will upload to the workshop webpage. And thank you.
Yes. Thanks to you for the invitation. And I mean, anything that you have questions, you can always contact us. We will share the slides with all the material and also the handouts. So all the material is available. And thank you.
Yeah, likewise. We've got the ChEMBL help address on the website, and any technical queries or requests to submit your data to ChEMBL, you can send in through there. Have a great day. Goodbye.
Okay. Thank you and have a good day. And since we are a little bit quicker, I think we can have a longer coffee break. And we'll start with the timing as it is in the program afterwards. So let's have a coffee break till 15:15. And during the coffee break, you can also put up your posters, of course. Thank you. This is Tak, nebo si to dej, jak ti to je dobrý. Dobrý. Jenom to asi zatím vypnu, ne?
Píš, že dobře, tak to vypni.
Okay, I think it's almost time to start. Maybe I can use this because we will be speaking about the protein structures. So I have one here. And maybe I'll leave it to our. So this lecture will be about the structural bioinformatics tools you can use for drug design. And the major reason is that the function of protein is given by its structure, which is like a definition in any biochemistry textbook. But what does it mean? It means that if you have a 3D structure, we are most interested here on the protein ligand complexes, of course, in order to make the ligand as a functional drug. But you can also be interested into how another protein is interacting with it, let's say antibody, which means we are also interested in how the protein-protein interactions come up. And that's also quite interesting things, how the SARS-CoV-2 spike protein is interacting with AC2 protein. Okay, I can leave it. Now I know. So I can leave it to you to try how easy it is to find the interaction place on this two pair of proteins.
Oh, I can skip it. It's too complex for me already.
Yeah. But we can also find out how the evolution plays out, because the evolutionary principles are now used to basically generate a new protein structures and to give up with the protein structure from the sequences. As I will be speaking in the next session about the AlphaFold logic. And it also gives us the shape and electrostatic properties, which will give us the information about how the protein actually works with its substrates and how it changes the ligand by the electrostatic interactions basically within the active site and how the reactions plays out in this case. And of course, that's how the catalytic amino acids and how catalytic place looks like. And we can use this information in order to generate the structures these days and how to generate a new products, basically. So the good thing about the structures is that we have them. And we have them in structural databases. Because currently, this century is the century of life science data. Because we have the data on almost everything. It's just hidden somewhere. You've already seen the data about the biomedical information from ChEMBL. I will be showing how the structures of protein look like. But we, of course, have sequences of amino acids in UniProt. We have the European Nucleotide Archive in ENA. When we want to see the sequences of the SARS-CoV-2 and how it changed, you go for GISAID. We basically have a data stored somewhere where it fits. And for protein structures, we basically go towards the protein database. But there is plenty of other databases. This is just a shortcut. There is not all of them, of course, because it's impossible to put all of the databases which are there on one slide. But there is one major database which rules them all. And that's Protein Data Bank. It's a combination of several databases, several places. So here in Europe, we interact with the Protein Data Bank in Europe, but the major one is RCSB database in the US, and there is also for the Asia and Australia, the position database is the PDBG. But there are not only the data for the proteins, there is data on on things. This is fresh. This is what I did this morning. So I haven't slept much. But currently we have over 200,000 structures in Protein Data Bank. Most of them is done via X-ray crystallography. Means you need to make a crystal and ray via Röntgen of radium in order to get them. And it's still growing by approximately 6% per year. The brackets are basically the change by the last year. Nuclear, the another way how you can make the structures is nuclear magnetic resonance. And this actually plateaued. The growth in this method is really slow these days. It's only 2% of growth. And the major reason is that the NMR is usable only for smaller proteins. And currently we are in the revolution using the cryo-EM. And the cryo-EM structures are the ones, that's the method which can solve large protein complexes, which are important for their functions. And you can see that by 1 year, it's almost 1/3 of the database is increased. And that's actually slower than it was a year before. So the cryo-EM structures are now taking quite a lot of stuff. Their limitation is, for instance, that you cannot do a small proteins with cryo-EM, because you need to have something bigger to be able to see it on the surface, because this is done on a surface of some graphene layer, and you are looking for some larger blobs, and then you are raying the electrons on them in order to get the structures. So this is good for larger structures, and we are starting to get a really nice resolution out of that method. Most of that are proteins, but we have there also nucleic acids and the nucleic acid protein complexes by itself. So this is quite nice. The protein data bank also contains some other biomolecules, and for us important is the structures with ligands, and basically 20% of this database is with the ligands in it. Of course, not all ligands are useful, because there can be like crystallization agents and so on, but there is quite a lot of ligands in it anyway. And it comes with a little problem, because formerly, when the PDB was 50 years ago derived, well, 50 or 60, 76 is the first one, 50. Then the data format which was used was PDB format. And this format was prepared for the Fortran computers, and therefore it had a specific weight, and you basically get individual positions on the line had some meaning for the structure. The problem with this format is that it was not extensible. And since 2001, PDB consortia basically started to look for a new format, and current new format, which is standardized, is a PDB X MMC format, macromolecular crystallographic information file format, which is used with the vocabularies, which is extensible, and it's much more descriptive about the data in it. The problem is nobody is using it. Yeah. And there will be a little problem next year, because for individual small molecules, we have in PDB 3 letters code for individual amino acid and for every ligand which is there. Okay. And they are running out.
My tady nemám bezdrotový sluchatka,
and they will be running
s tím
out
občas
this
trošku
year,
zápasy.
according to the people from PDBE. So please start using MMCs, please. I know it's not easy, especially in molecular dynamic simulations, but yeah, we can do it as a community. I hope that in next 10 years, we can finally move towards a more fair format. But whatever. Speaking about these statistics, we can see that it's growing quite nicely exponentially, and it's growing still year after year. And the major organism which is there, and the structures for the major organism is, of course, Homo sapiens, because we love to know something about our structures. The second one is small mice, which is a new thing, because 3 years ago, when I was running this presentation previously, there was E. coli as the second most typical organism. So the mice is probably moving forward. And for the enzymes, it's quite a lot of the structures of the proteins are actually enzymes in it. The interesting thing is where the structures are published, and actually the majority is not yet published from that. Yeah, so and then the JBC, JMB, and biochemistry are the 3 most typical journals from which the structures are basically generated. And when we are speaking about the structures, the most typical way how we are speaking about the quality of the structures is their resolution. And in general, basically 2 angstroms is the typical resolution, which is currently making the peak going downwards on both sides. So the most, I would say that 2.5 angstrom is basically a resolution which is important for the drug design, and the majority of structures actually have it. So this is quite nice for us. The PDB has, individual PDBs have their own web pages. Here is the PDB-E in Europe, and it basically gathers you the information about the function of protein, ligands, validation, structures, and more and more information. In RCSB, you get basically the same thing, but you get also quite nice genome viewer and PDB coverage. And there are multiple other structural databases coming from that. But I would specifically speak about this one. And that's Protein Data Bank knowledge base, which is aggregated view for individual UniProt IDs. Because PDB itself is built over individual structures which were analyzed, but you can have thousands of the structures for the same protein. Actually, for the lysozyme, because it's so easily crystallizable protein, which is used in any possible way, unless you are me again, because when I was trying to crystallize it as a crystallographer in training, I did not succeed. Again, one of the reasons why I'm a theoretician now. Then the lysozyme is crystallizable from almost everything. And therefore, there is more than 1,500 of the crystal structures of the lysozyme in this database. But most of them are actually equal. Okay? So it doesn't make any sense to look into individual structures per se, but it's good to aggregate the view and try to find out, okay, so individual structures are covering the structure of the protein, how much? And which structure is actually the best? What ligands I can find? And what is the major interaction protein, for instance? And are there any specific annotations about the active site, channels, antibodies binding, whatever? Okay. All of that is now possible to gather on one page, and then you can look in it and even make a 3D view of superposed structures. So we basically can look into that specific structure and to see whether there are 2 or more specific conformations. Like if you have a transporter, the transporter can be outward facing or inward facing structure. So you can see if there are these 2 different structures in that. That would be clustered. Yeah. So, and similarly, you can look whether there is one specific spot for the specific ligand, or if there are multiple places. For instance, for the kinases, every kinase has a place where it binds the ATP. But are there any others that can be used for that specific kinase? That's the things which you can get from this database. And of course, those were the experimental structures. But I would like to have to go forward a little and to speak about these 2, which is the computed structures these days, because there is almost any structure, every sequence you have, you can now make a structure. You can now make relatively easily a structure of it. I'll speak about it a little in the next part. The structure by itself, gathered from the experiment, has only the position of individual atoms. So the position of the individual atoms are, of course, important, but it's not gathering you the knowledge. The knowledge comes from the annotation. The annotation is telling this is this type of amino acid, which can be mutated. This is this amino acid, which is binding towards the ligand. This is the amino acid, which is on the surface of protein and therefore accessible for the, let's say, antibody. Yeah. For instance, in the spike protein, to know what amino acids are on what part of the structure actually gives you the information about whether the antibody would bind even into the structure when there will be these specific mutations. And it's quite nice to see the individual notations one by one over each other, so you can compare to each other and to see, okay, this is where the ligand binds, and this is where the most mutations are happening. So will it change when the ligand binds? Okay, so these are probably the resistance mutations, because they are changing how the ligand can bind. And this 3D bionodes from the Spanish elixir node is actually giving you that. You get the structure and all of the annotation for individual amino acids in its section. So that's the structure, and the annotations coming from multiple resources. But what we actually can see within the structure, we can find some kind of a function, and there is a little problem with the function definition, because what is the function of the protein really depends on the context. How you ask, because you can look for the function annotation by, for instance, for the enzyme. And in that case, you can use the enzyme classification to say, okay, this protein is oxidoreductase, host substrate is donor of hydrogen, what substrate acceptor type, and here you can have the annotation by the EC number. It's used by biochemists for half a century already, so why not to use that? But this annotation will give you only the information about what is the substrate and what will be the reaction of that specific protein sequence. But it's not connected to where exactly in the structure will it happen. Yeah. Another similar annotation, which is widely used by the experimentalists, is the gene ontology annotation. In this case, you have the information about gene, again, the gene sequence, and it's, again, quite generic one about biological process, and it can be even that generic like the cell growth. Well, that's majority of the genes will affect the cell growth, especially. Or quite specific about what type of the reaction will it speak to. About the molecular function by itself, so generic again, it's enzyme, it's non-structural protein, whatever, or quite specific about that it's hexokinase. And the last part of the gene ontology is where actually it will be. So it's localization of where the gene product will end. But again, all of those are quite generic ones, but still it's quite important for us to know them from the sequence. So there is multiple different possibilities how you can gather the information from new sequence. The first one is to use the sequence by itself. But from the similarity, you should be able to find out the function. That works a bit. But, for instance, if I'll take cytochrome P450s, which are the enzymes that are metabolizing the drugs and hormones by putting the oxygen specifically on some specific place on the molecule. Frances Arnold, the Nobel Prize winner, took the cytochrome P450, and there is the heme moiety which is making the reaction, which is connected by the cysteine amino acids. She changed this cysteine into the histidine, and immediately the cytochrome P450, which is oxidase because it adds the oxygen, started to be reductase. Yeah. So by one change of one amino acid, you can do quite a lot if you know where to do this amino acid change. And another problem with the cytochrome P450s is that while their structure is quite compact and quite the same, let's say all over the family, their sequences are not. They can have like only 40% of the identity and still be doing similar reaction. So it's really hard to do from the sequence automatically gather what will be the reaction, for instance. Okay, you have the structure. So another possibility is to use the structure to look for which structure is the structure I have and what this structure can mean. Because the structures are more conserved than the sequences are, it's more informative about what the molecule probably would do. So going via the CAT database, for instance, which is sorting the protein families by their structures, it's possible to look into individual families, and you can say that usually the same family, the same fold, usually have quite similar activities and quite similar reactions. You can look into the individual amino acid sites for specific amino acid positions. And you can look for the active sites. But again, speaking about the kinases again, the ATP is so flexible molecule that the binding site for the ATP in individual kinases has some similarity, but it's not the same. It's quite varied. So even the same molecule can have quite different multiple binding sites. But there are other possibilities, like what about not to use just one sequence, but to use the phylogenetic analysis to use all the sequences which are similar, and some of them might be annotated and therefore give me the information about the function. This is also quite useful for analysis of what amino acids are actually conserved, because they are usually conserved for a reason. There are some specific motifs like helix turn helix motif, which can give you the information about where the structure will bind to the DNA, and therefore where the molecule, whether the protein will be binding towards the DNA as, let's say, nuclear receptor. Some ligands can have similar sites within the another crystals. So for instance, for that, it's quite nice to use the PDBE knowledge base to look for the ligand. And sometimes the active site is quite buried, so maybe the channel leading towards the active site has some function. So there are multiple ways how you can see and look for the function within the protein. So that's the reason why it's important to look for the pattern detection, to see the patterns which are in the proteins and for the protein amino acids. And the major reason why to do that is that currently we have plenty of structures for the individual protein families. This was not possible in the past, but now it's almost impossible to look only to one structure to gather information about what the specific protein. And it's possible to look for, let's say, here testosterone and look for what are the amino acids around and gather this information to see all around, and even to find some binding sites via this possibility. But usually we are looking for the active sites because we are looking for where the molecule of our ligand would be binding. And as I've already said, there are several possibilities like looking for the size, and that depends again. We are looking for this large site or small one, or small molecule of the antibody. So in that case, this one is some protein-protein interaction prediction, which with the AlphaFold actually is getting attraction these days, and it starts to be almost reliable. Well, when I was doing it, it was nonsense. Or small molecules to see where the molecule might be binding. to see some hydrophobic patches, charged surfaces, to basically looking for that. And of course, now we have the time of the machine learning. So here is one example from Charles University and Czech Elixir. That's the P2 rank software, which is run by David Hoxha's team. And this they are basically looking via the all of this information taken together by machine learning and trying to find out where are individual pockets and what can bind into them. And this I've tried on the cytochrome P450, which has one large cavity inside the structure. And of course, it predicted it quite nice. So yeah. But here we've been searching a lot for the channels, which are basically leading either towards the active site or via the transporter from one side of the membrane to another. Started with the caver. Basically, the question was to find a way from one active site towards the outside of the protein by some kind of journey. That's now done at Loschmidt Laboratories by the team by Professor Damborski. And basically, we are looking via the Dijkstra algorithm, or they are doing it as well, via the Dijkstra algorithm to find the closest way how to get out of the any spot. We have MOL, which is basically doing similar thing, but it had transformed the atoms into the Voronoi diagram, which basically is looking what's in between the atoms. And now we are basically searching in those ways in between the atoms. And we have this MOL online, well, which is here. Basically, what you can do is to upload the structure or to look for the structure from the PDB and try to find out the tunnel in it. And for the individual tunnel, we gather the information also for what are individual parts of the channel, what are the properties there, what are the amino acids there, and therefore you can look for their for the leading residues. Can tell you something about the function. And of course, we use that for the analysis of the PDB. So we've used it for the analysis of enzyme structures. And we took all annotated enzyme structures from PDB, which was like 4,000 structures in 2014. Now it's double the number. And we calculated from the active side, we calculated the channels. And we were basically interested into, is there any channel at all? Or isn't it? And we found out that actually at least 60% of the enzymes have some kind of a channel leading towards the active site. And this channel, we take at least 15 angstrom. It's quite large in it because the shorter ones can be just a cleft leading towards the active site. But these were the really 15 angstrom is not really short distance when the small molecule is like 5. The middle channel was even twice as large, or almost 30 angstroms itself. We found out that most of the channels are actually polar, and especially the bottlenecks is made by the aromatic amino acids, which were like phenylalanines or histidine, which can basically swing in, swing out. So it makes sense that these amino acids are basically opening and gatekeeping the active site. And from this analysis, we actually can say a little bit about what the structure of the protein usually looks like. So if you have a structure of the protein, then inside the protein are aliphatic amino acids. They are basically those ones which are making the structure stable. If you have a catalytic site, then in the catalytic site, you usually have one one or two specific amino acids, which are usually those charged ones or aromatic ones, because those are running the reaction. They are giving the electrons or taking out the electrons or hydrogen, vice versa, in order to make the reaction going. Then the internal part is usually only polar or aromatic. Especially the aromatics are for the bottleneck. And if you are on the surface of the protein, then the surface of the protein is usually made from the charged amino acids, because they help the protein to be solubilized usually. And usually all of those enzymes which were crystallized are the proteins which are solvable. Yeah. So from this analysis, we can say that those aromatics are usually in the structure for some kind of a really nice gatekeeping property. We collected the channels into our channels DB. There we have already the protein channels which we've reviewed from the literature, usually from the papers that cited us or Karel. Then we also have these tunnels running from the catalytic site atlas. So you can basically look into the tunnels that we used for our analysis. And then we thought, okay, those were the catalytic sites which were annotated. But usually, if you have in the structure some cofactor, then this cofactor usually is also site of the reaction, even if it's not annotated. And therefore, we have there also tunnels running from those cofactors. So for instance, if you have a heme, like in the cytochrome P450s, or if you have flavin adenine dinucleotide, we have them. And the tunnels leading from that side. And the last but not least are the transmembrane pores going from some porins and some transporters as well. Now the data are there calculated with mole, but I hope that within 1 month we will have the data calculated with Karel as well. So the times they are changing. So that were the channels. And now let's speak a little bit about the specialty servers that can help you with the other issues you might have. Yep. 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 Yeah, the question was, because you probably couldn't hear the people online. The question was, what about the flexibility of the channels? Both us and Karel are taking into account the flexibility by running molecular dynamic simulation. And both Karel and us, basically, you use the snapshots from the simulation to look for the channels in those individual snapshots, and that's the same for both softwares. Yeah, but you have to download it, because run the trajectory on the web, that's a lot of calculations. But from the usage statistics, someone is doing it on our mole server, because we got a quite nice spike in 2020. That was basically, but that someone was analyzing the trajectories. So it was quite a spike in the usage of the server. And since we are storing the calculations on the server, basically found out after a while that we had to buy a new hard drive to store the data, because we did not anticipate it that much of the interest. But for that, we have the standalone application that can be downloaded and run on your side. And it's quite quick. So that was for the channels. And now let's move towards the special parts, a special type of the proteins. And I would speak specifically about the membrane proteins and disordered proteins. Why to speak about the membrane proteins? Well, I've already said it in my introduction that more than 60% of drug target are actually membrane bound. So we have to deal with them. Previously, it was hard in that sense that there were no structures for them. But now with AlphaFold, we have even the structures for the transmembrane proteins and membrane-bound proteins. So now it's possible to do the analysis. And then there is a question about the composition of lipids in different parts of the human cell or any cell. And basically, you can see that if you are close to the center, then the membrane is more filled with the common lipids like phosphatidylcholines. But more and more, if you are moving towards the surface, the membrane is filled more and more with cholesterol, which makes the membrane less permeable. Yeah. So the inner membranes of the cell are usually more permeable for the molecules than the outward one. Makes sense because usually the cell does not want to interact that much with the surroundings. But the protein content is also quite huge in different membranes as well, especially quite different in mitochondria. So since we have these proteins, but we don't know from the structure where the membrane is, there is several servers that gives you the information. And the one which I like the most is the server from Andrei Lomize, which is called Orientations of Proteins in Membranes. It's called OPM server. And it's actually the data from this server is now all added into the RCSB webpage. So it's possible to gather this information there as well. The good thing about this is that it's really quick implicit solvent calculation that gives you the information and orientation of the protein on the membrane. And it gives you the information also about which type of the membrane it probably is interacting with. Not always true, but they were replying when I was saying something is not completely the best. But the good thing is that you get the information which part is membrane one. Actually, do you have the AC2 protein structure? Okay, so I've lost my 3D prints. No, the another one. Okay, so this AC2 protein structure, this AC2 protein is actually membrane bound as well. And there is the SLC transporter connected with it. And from the size of this SLC transporter, it actually is possible on the structure to see where the membrane is binding, because there is so-called lily-pad structure on the structure of the protein, which shows that some parts is basically squeezed, and on both sides of the membrane, it actually is enlarged. And these lily-pad amino acids are usually aromatic ones. That's also quite important. And if you compare that towards the SARS-CoV-2, you can see that where this is attached to the membrane, it's not really easily recognizable because it's much longer stem. And if I compare, it basically says that it's interacting like this. And this part is actually quite flexible and helps the virus to see for their counterpart. So this is also some information that comes from the structure if you analyze where it binds. Another thing, which is quite funny, speaking about the structural bioinformatics, and that's speaking about the structures, the protein structures which are invisible because they aren't possible to have their crystal structures. And they are so-called intrinsically disordered proteins. These proteins are full of charged and specifically polar amino acids, and they serve as some kind of a flexible chains that can have multiple different regulation roles, like molecular recognition. It can bind towards one protein, towards another, and bring them together. It can help the molecular assembly. So for instance, if I will look into the structure of the SARS-CoV-2 nucleocapsid protein, there are two specifically quite nicely crystallizable domains, but there is a large loop which is completely unstructured one. Right? And this unstructured part is helping somehow assembly the SARS-CoV-2 RNA structure together towards inside the viral capsid. They can lead towards the protein modifications, and they are quite entropic. So they can serve so-called entropic hourglasses. It's quite a funny name, but it basically helps the inner counting of time of the cell. And the best way where to find them is in a DISPRO database, where you can find them from individual organisms, and depending on what type of that is, what are their regulation functions and whatever. And there are multiple structural generators for those intrinsically disordered proteins coming from the, let's say, NMR ensembles. There is possibility to look for them and to basically try to fold them somehow, because when they are quite oftenly when they are in the context of another protein, they can form the structure. And one typical example of that is one of the most typical cancer protein, and that's protein P53, which does not have one specifically structure. But in PDB database, you can find it in multiple different structures, binding towards different targets. And this protein is regulating one, and when something is wrong with this one, usually it's cancer. So, yeah. So, take-home message from this part. There is an enormous amount of information on structures available. The structures are as good as the annotations are. There are two major databases to go for the structure, PDB, for the sequences UniProt, and they are connected. The structure, or no structure, defines the function quite nicely. And quite often there are channels, things about that, if you are specifically in a low mode or no. And disorder may be also quite important. And so far, we haven't seen it. So it's probably quite interesting area to analyze. And with that, I think I've managed it completely, almost specifically on time, before my previous second lecture. So do you have any other questions towards this part?
Regarding the mole and caver, so
yeah.
for ion channels, for example, which are also have their resting states and then liganded states, desensitized states. So have you been able to follow the pathways of ion flux or that, like the blocking?
Well, the blocking you can see quite easily of the ion channel, because if the structure is blocked, then you can see no channel. So that's easy. The problem is from the static structure to find out where it will be moving. And that's the only thing which we came up with is basically the dynamics. So if you have the dynamics of the structure, then you can see how this, how the tunnel or channel is basically briefing and how much place you have there.
By the way, just to follow up, like I understand from like when you have an enzyme prosthetic group going to the surface,
yes,
both of this you define. When you have an ion channel, how do you define like what is a channel from where to where?
If it's membrane channel, then it's easy because we use the OPM. So we define where are the parts of the, where is the membrane. And then basically we are looking for one way from the middle of the protein usually, and how to get to the middle of the protein on the another plane. So that's our leading for the mode. Yeah.
We have also some data for the average diameter of the channels. Like if the diameter is to accommodate, I don't know, water molecule, or like, you know, how wide the channels are, or the balance to the arc.
The statistics for individual channels and whether they are defined by individual ligands, we don't have per se, but we have it when we've done it for these enzymes. We were expecting that for instance, for the same type of the enzyme, we would have the bottleneck, which is the smallest place, would be on the same place, in more or less on the same place or along the channel. That was not true, because then we ran some molecular dynamics simulations and we see that even if the channel is seemingly too narrow, it can open when the ligand is moving. It can be said like a peristaltic motion in intestine, that it moves towards more and more towards the active site or more and more outwards, depending on whether it's attracted or repulsed from the active site. And the reaction can change this preference. So for instance, for the cytochrome P450, we were able to analyze that one channel can be analyzed that it was coming from one channel towards the active site because the substrate was attracted to that specific place. And then when we change the substrate towards the product, then the product did not want to go via the same way, and it selected another way to get out with lower energy. So it was possible to do that, and it's possible to combine, let's say, mole and autodoc vina, but which is possible doing it on the desktop side, let's say. But I know that Caver has the CaverDoc software web server, where you actually can upload the structure, the ligand you are interested in, and look for the channels, and it will dock the molecule along the channel to gather you information and give you the energy profile of this road. So it's quite useful, I would say, even though it's coming from them and not from us. Okay.
What is the average resolution of cryo-EM structures?
Better and better. The resolution of cryo-EM 10 years ago was, if it was lucky, it was 5 angstroms. Now it's 1 and 1.5. And it's getting better and better, even for the large structures. But with the overall resolution of overall protein, it's always a little bit shaky, because the resolution is important where you want to see at that specific point. And that's not always the true that in the center of the protein, you would have the same resolution as on the surface. Yeah. One reason why you usually have a difference is that the surface of the protein is usually more flexible, which is actually hindering the AlphaFold and all the other protein-protein interaction predictors, because it's much more flexible, much more flexible in the structures, and the structures are not complete. Yeah, so it's much harder to basically predict something binding there. For the crystal structures, usually when you have the ligand bind towards the active site, then it usually freezes out the active site, so it has a nicer resolution than the rest of the protein. But it's necessary to check it out, because it can happen quite often that the resolution of the ligand, the electron density within the active site, is not good enough. And quite often, when you are looking into the active site in detail for the electron density, it can happen that basically the crystallography puts the conformation of the ligand in one way, but there is some unattended electron density close by, which can be water, can be iron, can be counter ion. Sometimes quite hard to catch what is there and how it can change, for instance, the reactions. No. Okay, is there any question in the Zoom? Is there any question? No question, and no question in chat?
No questions in chat.
Okay, so let's move.
Maybe wait 2 minutes because we have lagging sound.
Okay.
But I think nobody is writing anything, so we can continue.
Okay. So, without further ado, let's move to the last lecture of today. I hope you are as happy as I am. And that will be about the alpha-foldology, which is to state that after the release of AlphaFold 2, there has been so much of effort about usage of the machine learning for the structural biology that we, together with Marian Novotný at Charles University, decided that it deserves a new name, looking for what the AlphaFold is making towards the structural biology, and we name it as alpha-foldology. So that's a little bit this explainer what does mean. So in this lecture, I will be speaking about how the protein structure prediction actually worked, what happened in CASP14, and then I will be speaking a little bit about the AlphaFold itself, how it's working, and then how it can be used and where you can use it, and what are the limitations. So in 2020, in November 2020, there were nice informations like it will change everything, DeepMind's AI make gigantic leap in solving protein structures, AI triumphs at solving protein structure, and so on. But it's journalism, and it reminds me a little bit of this, where we have the complete human genome since 2001, and just this year, just last year, there was a new science paper in April 2022, where it says that it's filling in the gaps and closing in on to complete the human genome. And it's still not completely done. Yeah, so sometimes if said that something is completely solving everything, it's nuanced. Yeah, we've already stated in the previous lecture that when we know the structure, we can understand the function better. In this case, when we have the structure of the HIV protease, we can see, or this is kinase. Sorry. We can try to make some drug interacting with it. We can understand how the biology works by analysis of the structure of this nice ATPase. And the problem is that the solving of 3D structure experimentally, even with the cryo-EM revolution, is still difficult and still expensive. And if we compare that in 2019, we had like 160,000 protein structures in the PDB database. Now, okay, now we have 200,000. But let's state it, 20,000 of those are actually from COVID. Yeah. And the number of sequences is going over the roof by 300, 600 millions of sequences. And this is just the millions of UniProt sequences. It's still going on. So the question always been whether we can use the sequence in order to make the prediction of the protein structure. Do you read? Do you read? No.
No. No sound still.
But it's green.
Yeah, I do.
Okay, so it was batteries. Cool. That's easy. That's doable. So in order to make the validation, you basically need the crystal structure or the experimental structure to validate on. But if you have the structure, then it's quite easy to cheat, because you can make the structure basically by taking the structure from the experimentalist and then say, okay, I made it by the theory. So in order to take these things apart, the Professor Mould basically designed a competition which was called Critical Assessment of Protein Structure Prediction, which is run since 1994 for prediction of the structures. And it's done in a way that basically they ask the experimentalist, okay, if you have a new structure to be released, give it first to us and give us the sequence. So we will give it to the theoreticians that will run their prediction. And this prediction can be afterwards evaluated whether the prediction is similar to the experimental structure or not. Yeah. So this is how this thing works. Later on, there even came up a spin-off of this competition, which is called ChEMBL. And this one is basically using the fact that the PDB database is always uploading new structures on first day, while the sequences can be available during the previous week or two. So they are basically running a small CASP automatically each week, and usually the automatic servers are compared with that, and automatically each week it's automatically updated and analyzed by machine within the ChEMBL, and the teams can make and predict new structures on a way. So, but the major one is CASP, and in CASP basically that's how it looked like in the past years. So it was the first ChEMBL was really that the quality of the structures which were difficult was really hard. And this GDT-TS is a measure basically looking into how much of the atoms are close by to the experimental one via 1 angstrom, 2 angstrom, 4 angstrom, and 8 angstroms. So in order to get 100, you get to have all of the atoms within 1 angstrom of the position. 80, 75, 1 angstrom is not enough, 2 angstroms basically, and so on and so on. So at the beginning, even the 4 angstroms was too much. The 8 angstroms were too much for the difficult ones. For the easy targets, usually at that time, where the easy target was the structure which had some kind of a template, that was easy, even at the time. And then it made deep the first year, but then basically it stalled for basically for a decade or more. It stalled because nothing was changed. There was a tiny differences between individual year and year. It was moving this average a little bit higher. Then in 2020, the first AlphaFold, which was at that time calculated completely closed inside the DeepMind, entered, and it immediately got this. So a huge increase. And there were quite a lot of noise about that, because it was like a private company using so much computational resources that no academic lab would have any meaning to gather that was able to calculate those. We are doomed, because it's basically the thing about the computational resources. But then in 2022, it was 1820 was that the new version AlphaFold 2 won in a way that basically everything was between 1 and 2 angstroms. So almost perfect. Of course, there were some structures which were down, but not that much. But interesting thing was that even the structure, even the field without the AlphaFold get to the previous version of the AlphaFold, and was a little bit better than the previous version of the AlphaFold. And that's thing which basically happened quite nicely because they realized that it's possible. And they gathered a little information about how it is done. And they were able to basically mimic it. And last year, there was the CASP 15. And now, the first method, which does not use the AlphaFold, is here. All the rest is using AlphaFold as a basics in order to basically build upon the AlphaFold and use the AlphaFold model and just tune it depending on their different approaches. So there are several AlphaFolds which you can use quite easily. And the funny part is that this graph is now pointless because all of those servers are at that level of resolution. So yeah, that's the reason why they switched for this one. Yeah. So how does a good prediction look like? Like this. So you can see that basically you can follow even the side chains quite nicely towards each side. And the worst prediction of AlphaFold in 2020 was this one. Really, what a mess, I would say. The only thing is different that this helix moved. That's it. Yeah. And even the prediction of the individual local details, like this ORF9 protein in COVID-19, basically are moving all around. So it's quite good. How does it do it? So we know that the AlphaFold, as an input from the user, asks for the sequence. But this sequence is immediately moved towards two different parts. First part is looking for the genetic database and trying to find out this multiple sequence alignment. And the second part is looking for the templates for the structures. These two things are basically, this generates the database, the sequences generate multiple sequence alignment, and the templates are generating the pair representation, which basically gives you information about how individual amino acid is interacting to each other in the structure. These two representations are then mixed in a software called EvoFormer, which basically tries to optimize the representation of both and tries to come up with one single representation, with one optimized pair representation that afterwards generate the structure in a model. And this structure is then validated by predicting a predictor of the quality of the structure, which was tuned by machine learning as well. And this is actually one of the major parts which is now really useful on the AlphaFold by itself. Then it took the high confidence structures and starts to recycle it again, to add it here to the structural motif, to optimize the representation of the sequences, and to run it again to gather even better structure. So why to use multiple sequence alignment? And how is it really necessary for the structure to be formed at the end if you run the structure model? Yes and no. The multiple sequence alignment seems to be working as some kind of a pre-filter of good structures. And then those good structures are serving as some kind of a templates for the generation of the structure at the end. So if you have small amounts of the sequences at the beginning for the multiple sequence alignment, then the quality of the structure which you get at the end is deteriorating. But after, let's say, 30 sequences, you get basically almost the same quality even if you are going for the hundreds and thousands of the sequences. The training they did for their model was based on PDB database, but it was based on specifically a good quality structures from the PDB. And they trained to predict the PDB itself. And basically they cropped the structures to basically 258 residues for no obvious reasons other than it's somehow multiplied by 2. So it was probably good for running on a tensor processing unit they had. And then they enhanced the accuracy by noisy student self-distillation. So they basically took the structures, run it through, and then find out which ones are good ones and which are not, and use that for training the model further to filter the high confidence from that. So basically that was to learn the model to find and tune for a really good structural structure mix, let's say. And therefore, it was possible to use that for even the unlabeled sequence data, which were coming from the multiple sequence alignment. And then they've even tried to do the BERT transformer to mutate individual residues in multiple sequence alignment to predict masked elements that might be in multiple sequence alignment. So the Iverformer module is combining all of those information together in this series of things. So first you have a multiple sequence representation, and you add the information about the pair representation. So you get information about what in the sequence alignment should be where on rows and on sequence on columns. And then you transition it back towards the pair representation, because the pair representation is at the end defining which amino acids, what will be the distance of individual amino acids to individual amino acids. Yeah. And then you use triangles, not pairs, to define the structure. And that's another nice part about this representation, that previously the convolution was done only on pairs. But here we have the triangles, and you have the triangle inequality that gives you the information, okay, if these two are that size, then this one has to be larger if there is another distance. And you basically use outgoing edges, incoming edges, and starting node and incoming node. And basically these triangles are giving you more information about to generate the more proper and more detailed pair representation of the model at the end. And if you have more defined pair representation at the end, means that you at the end would have better structure coming from the pair representation via the structure model. And the structure model basically takes the pair representation and then at the backbones as just flying points in space. Do not connect them together into the backbone per se, but basically to leave them as a freely rotatable blocks, where the blocks basically were put afterwards the position of the side chains, optimization of the side chains, and only after that you connect back towards the main chain. The reason for switching this from the original way, where in case of the template modeling, homology modeling, you basically took first the structure from the template and you put there first the backbone, and only on backbone you put individual side chains. And here you are doing it reversibly, because you first put there the side chains and then let them optimize and then click back the backbones because that allows you a little bit better optimization of the final structure. And the last part, molecular dynamic simulation. So typical shake-up. So what is it possible to do? One thing which is quite interesting is that even though you are predicting only with the original version of the AlphaFold, it was possible to predict only the individual channels, individual chains. It still got the structure which was in a multimolar in the experiment. And the major reason for that was that it was in this structure also in the original structures which were similar towards the from which it was built, basically. So after 2020, once the AlphaFold 2 came up, in 2021, in summer, there was a release of this AlphaFold database, which is run now at the EBI, from which is also ChEMBL. And the protein structure database basically allows everyone to basically gather the information for the sequences that were solved with the AlphaFold. It started with the 48 organisms, including Homo sapiens, and predicted structure for all of the proteins coming from the human. And it also added the predictions for the global health proteomes, which means usually the infection bugs. And finally, it added the data for the SwissProt and later on for UniProt. And it was connected back as well. So now when you go for the UniProt, in every UniProt ID, you get the structure of the protein listed there as well. So now we have full prediction files from the SwissProt, and there is approximately 200 million plus structures available. So individual structures look like this. Here is a mole star coming from Brno. But here are the individual colors stating the quality of the structure. And depending on the quality of the structure and the color there, the model confidence is using this predicting quality of that specific part of the structure. And from my own experience, when something is dark blue, therefore is very high, the structure stays put. It will be there. If it's light blue, it means at the end that the structure can move there. So for instance, if I will look into the transporter, then the majority of the helices which do not move are dark blue, and those ones which are changing within the transporter movement, they are usually either confident, therefore cyan, or low, which is the yellow. And that's one interesting knowledge which came up from this AlphaFold as well. And this is that this predictor from the AlphaFold is actually really good predictor for the flexibility of that individual protein on that part of the protein. And it also gives you this, this graph that gives you the information which amino acid is interacting to which another. And it can give you the information whether the domains from the protein, whether they are aligned well, or whether some part can be shifted from one side to another and so on. It's interesting that if we have this data, it's interesting to look for the predictions of the human proteins. And the resolved protein humans have really good confidence in them. And there is only a little part which does not have much of the which have lower confidence by the prediction. Which makes sense because those are the structures which were resolved, and those are the structures which have the structure already crystallized. So it's quite nice that it gives you the information that those are done. The unresolved proteins from human are really tiny, have high confidence, and quite a lot of those actually have quite a lot of flexibility in them. Which again, there is a reason why those structures were not resolved by crystallography. They were too flexible. And when we took the human in all, then approximately 50% of the human is really nicely resolvable. So quite a lot of human proteins actually are with some defined function, some defined structure, especially if you take into account also those confident ones, as well those flexible ones. But there is 1/3 of the human proteins which are quite flexible. But still, one needs to be careful. So, for instance, these are two proteins which are both from the cytochrome P450, and you can see that some of those are not really well resolved, and that's coming from quite a lot of missing sequence. So the structure still can be as good only as its sequence is. So if something really big is missing in the sequence, then of course the structure will not be good. Also, another problem is with the AlphaFold that it contains only the monomeric proteins. Here is the Mycoplasma smegmatis ATP synthase, which is from the AlphaFold DB. And you can see that there is quite a lot of and we were specifically designing the compound as a drug for this specific protein. And the problem with the AlphaFold was that when I've aligned it towards the crystal structures of the another cytochrome, another Mycoplasma, not tuberculosis but smegmatis, it did not fit. And when I've looked for this Swiss model, it fitted perfectly. So if you have a really good template with the multimer, then the template homology modeling is still better than the AlphaFold, because this is a relatively small movement of the protein chain, and since it's not taking into account the another unit, it does not end it well. So sometimes it's necessary to take some caution. And after the AlphaFold, well, it was quite funny. During the last November, I went for the Elixir conference at the EBI. And when I landed there, Meta AI released this even larger database of protein structures, which was metagenomic proteins. They contain like 600 million structures. And the reason why they were able to do that is that they did not use the AlphaFold, which was basically based still on the deep learning over the protein structures, but they used a large language model for protein structures. And they've trained this they've trained this large language model similarly as ChatGPT was trained over the internet. But in this case, they've trained it over the proteins, and it's much quicker. Because if you know, have you tried to run with ChatGPT? Yeah, I guess so. Yeah, because sometimes it's quite good way how to get, if you are stuck with something, just how to get rid of that. It's really quick model at the end. And this ESM fold is quick as well. So it even allows you generate structure on the go. So I've tried what will happen when I've put here the folding on the fly, and when I've added there the name Andrew White, which is quite nice because it's short enough, so you can make a multiple versions, copies of it later on. And it's all of those individual letters are amino acids. Okay? For instance, I cannot use it with my name because there is unfortunately no amino acids called B. So, yeah, problem. But with Andrew White, I've put it there, and it immediately gives you the structure. And if you put there Andrew White 50 times over, it still gives you the structure within 10 seconds. So it's quite interesting what's happening during that time. And if you want to see this experiment is on my Twitter account, so you can find it out later. Yeah.
It's actually the representation of the protein they are using can be used with a language model.
I don't know the details. I haven't looked it up, I must say. But they on the ASM Atlas, there is a paper of the ASM fault aligned, so it's possible to look into it. Yeah. ASM Atlas, and here is the research paper on both Atlas and also the language model. The difference towards the AlphaFold is that the quality of the models is worse. But given the enormous quality of the AlphaFold models, in most cases, it's enough. Yeah. So if you want to get a structure really quickly, it's easy. But if you want to get the structure from the AlphaFold, the right one, there are multiple ways where to run the AlphaFold. One possibility is collapfold. There is a quite nice collapfold community, which is run by Socrypton on GitHub, and it's running on the Google Colab engine. And you basically select what you want. So, for instance, I want the complex, I want the batch, I want the AlphaFold 2. And it's in a Jupyter notebooks, which you run over the in a Google Cloud. And if you run one structure at a time, it's relatively slow, but what you get there. But if you want to go quicker, then you have to pay for access to more cloud resources. At Elixir, there are several ways how to run the AlphaFold. So, for instance, if you have the access to check Metacentrum, on Metacentrum, there is the AlphaFold instance where you can run the AlphaFold on Metacentrum. If you are more familiar with the Use Galaxy, then it's possible to run Use Galaxy EU, where it's possible to run AlphaFold prediction as well. And here, for instance, I've tried to make the structure of this nucleocapsid protein of the SARS-CoV-2, which is making dimers. So when I was doing it with the common version of the AlphaFold, it was no avail, because when I've added two copies, and I've aligned it across the dimeric interface, the chains were colliding to each other. So I've used the trick, and in the middle of both structures, I've put it a long disordered poly-N chain. And the trick behind the poly-N chain is that the asparagine is the polar amino acid, which is, if you put there a asparagine as a chainer, let's say, then you are basically putting there intrinsically disordered protein, because poly-N is intrinsically disordered protein, and therefore you can connect anything. pivot. So this is usage of the IDP knowledge onto the protein prediction structures. Yeah. And you get the structure with the representation, and you can see, and later on you can delete the poly-N chain, so it's easy. So what are the limitations of the AlphaFold? Well, the AlphaFold is currently only the start, and is already used in multiple different approaches. It's used for molecular replacement, it's used for the representation of cryo-EM structures, and so on. And I've taken this today, and there is like 2,700 research articles mentioning the word AlphaFold in their title or in the abstract. And it's even funnier is that 227 papers were published in 2023. We are at the end of January of this year. So it really is AlphaFoldology, I would say. So one of the first papers which started to use this, the AlphaFold, was for the tool for usage of molecular replacement for the crystallization. Because for the crystallization, if you have the structure of the protein, then it's much easier to generate the final crystal structure if you have a template to put into the electron density map. So the MRPARS basically is doing that. So the question is, are structural biologists and bioinformaticians out of the job market? Well, not yet. Because AlphaFold cannot do a lot of things. First part, it cannot do the point mutations, because the point mutation is not in the evolutionary part of the MSA. Yeah. So it cannot get it. Cannot be blindly used for drug design, because the structure does not contain any ligand. It's just pure protein. So if you have something in it, then some cofactor or something that is necessary, then you don't have it there. It cannot do the conformational changes of dynamics by itself. It runs, well, originally it ran only the monomer, not the multimer. It did not create the structures which would have some effect of post-translational modification, like phosphorylation. Again, it cannot predict good orphan sequences, and it cannot tell you anything about the folding process. Or can it? It can. So for instance, this is my study when I played a little bit with the minimal sequence code for switching protein structure function. This was done in 2009 by Patrick Alexander experimentally, when they found out two protein domains which were quite similar in the sequences, but they have quite different structures. One was triple helix, and one was helix with the beta sheets. And they've mutated one amino acid after another, and finally they found out this one mutation which switched from one structure to the other. Yeah. So AlphaFold, could it do it? I've played it, and I've took these sequences and I put it into AlphaFold to generate the structures. And eeringly, the all alpha was predicted quite nicely all the time, and all beta was also predicted nicely. The problem was with this one, when it was supposed to be beta, but it was beta only in 3 out of 5 tests. So it actually was able to find the switching point of these two. And it was able to give me where exactly is moving this part. So one helix was retained everywhere for 100%, and all other helices had a presence only in some structures, and beta sheets were found only in some of the structures, according to the sequences I've gathered. So I think that it actually can work with the point mutations. How good is this for drug design? Well, we are at the workshop for drug design, so that's the major question we are asking here. And actually, there is an analysis that AlphaFold actually predicts the hollow structures of the proteins. Therefore, those structures where would be bound ligand. The reason for this is probably in a way that when we were training it, we did not put the ligands into the structures, but the holes for the ligands were there in the training set. And as I've shown you in the PDB description, quite a lot of structures actually have ligands in them, 20% of them. So those ligands, even though they are not present there in the structure, they are present in the sequence around and in the organization of the amino acids around that specific spots where the ligand would be binding. So actually, it can be used quite nicely. AlphaFold can predict the dynamics because the AlphaFold can give me the information about which parts is actually dynamical. And there is now, we are a little bit further, it's possible to do a little changes to the conformational, to the multiple sequence alignment in order to tune the AlphaFold to generate the structures of individual conformers. So it's possible to use this now to map the conformational movement of individual structures. So it starts to be impossible to generate the conformational ensemble, not only the single structure, but the conformational ensemble of the protein, which seems to be interesting for the future. And another place where it's interesting to see for the ensembles is interestingly disordered proteins. And as we can see, when there is the interestingly disordered part, AlphaFold basically puts some kind of a strange jelly, right? Strange chains. Of course, the structure will not look like that. And before you start to do any docking or any analysis of the structure, you can delete those parts because these are the computational artifact. But at least you know where the flexibility of the protein is. And it's so good that it's important enough that it gives you the information about the individual folded intrinsically disordered parts. And if you compare it with the multimolar now, you can fold those intrinsically disordered proteins to make their binding sites towards the spot of that other protein. And this PLLDT score can be used as a measure of the flexibility of that specific part of the protein. AlphaFold, of course, can do now multiple protein complexes, and it gives them relatively nice in some cases. So it's getting better and better in this approach. It's not completely perfect yet, but it's getting there. And it can give you also the information and generate the individual, well, one trick how to make the multimer out of the single monomer. I've shown you with the long IDP linker, and you can basically take two proteins and try to convince them to be one, and it works. There. And by itself, it cannot do the post-translational modifications, but it can give you the right places, and there are another predictors that give you the information, for instance, where the sugar moiety will be. Yeah. And for the ligands, there is the AlphaFill database where you basically take the structure of similar proteins with the ligands, and basically you transform the ligand into the AlphaFold structure and do a little bit shaking. So it gives you the information where the ligands would be in those structures, and you can use that information later on. And as for the folding process, it can give you a little bit of detail of the folding process itself. Basically, if you are looking for the intermediate of the protein, how the molten globule would start to form. And basically, the analysis of this is that the multiple sequence alignment gives you good initial guess to find the right local minima, and then you use the structure module to locally descend towards the global optima there. So finally, AlphaFold gave us a huge leap in the precision of the protein prediction, protein structure prediction. It cannot be overstated that it was possible only due to the fact that all of the protein structural data are for 50 years in the open database called PDB. Because without this database, it wouldn't be possible anything like that to achieve, because there wouldn't be any data. And in PDB, there is a lot of annotations done already by previous and curations and so on. So without that, it wouldn't be possible. The important part was also the CASP competition, because it allowed the iteration and basically analysis how good the software is. And now the AlphaFold is publicly available. It can be used on Elixir Check resources or use Galaxy resources. And it has inspired a lot of uses and tools already. So basically, you can start from the AlphaFold and move forward. And it seems that the structural biologist will basically use the AlphaFold as a start when they will be starting with their journey. And with that, thank you for your attention. I have one last slide, and this is this nice tweet from Kristen Larsen, Lindolf Larsen. Just tell me again how the folding protein has been solved, because these are individual conformations along the journey of one protein. So at least we have something to start from. So back again. Do you have any questions?
We have one. You said that molecular dynamics is used at the very AlphaFold. So I guess as it's not deterministic, so it depends which force you use, how long you run, whether you take one structure, whether you minimize. So is this how does this affect are there some restraints?
From what I've understood, they are position restrained, and you just run the molecular minimization. And you run it with the Amber force field.
Yeah.
So it's really nothing really special. Are there any questions in the chat?
AlphaFold does appear
great prediction, but how much can we rely on IDP's prediction of AlphaFold?
It seems that the prediction of the IDP of this score seems to be quite good, because I haven't seen much better working prediction scores from my knowledge.
Here is the second one.
Yeah.
Some people publish survey and know if AlphaFold helps you on drug discovery project. Experimenting, and many won't know. A few days ago, some article published about AlphaFold and simple medicine and discover liver cancer individual using AlphaFold. So, what is your option? Will there be many cases like AlphaFold liver cancer every day? It's not a question.
That's a really long question. I hope that there is this Zoom running still. No, it's not. So if I understand it correctly, will there be drugs which would start their development with the AlphaFold?
Yes.
Yeah. Yes. Yes, because previously the problem with the structural drug discovery was that for quite a lot of those membrane-bound proteins, which were important for that specific drug targets, we did not have any structures. Now, with the AlphaFold, we have the structures of them, and therefore we can run the virtual screening on them as well. So I think that's the major effect, how the AlphaFold will affect and how the AlphaFold will enable structure-based drug design, which will hopefully lead further towards the drug candidates on those hard targets. Okay.
What is the fate of this CASP challenge? What is the future? Because if currently they achieve almost 100% accuracy, they should change something.
They did. I think it will be the best to show on the CASP. So the CASP 15 you can find quite easily on their webpage via just Googling CASP 15 and it will give you there. But its important part is that there is the doc presentation. And the CASP itself is also a conference where the people are presenting the results of individual parts of the CASP challenge. So day one, this is, there were basically the targets of CASP 15 on and discovery what was used. Then there were from John Bolt, there was the introduction to CASP 15 and Xavier Robin discovered ChEMBL. That's day one, basically. That was one. On day two, you can see that there were the description about the assemblies. And the problem with the assemblies is that quite often you don't have the full assembly of those proteins in the PDB. Because it's quite hard to describe whether the contact you are seeing is a real natural contact, protein-protein interaction, or if it's just a crystal contact. So that's one part about how to describe that. Another thing is basically invited lecture from John Jumper, that's the alpha. And there are the individual predictors and how they did it. And the first day, basically, it was question about the ensembles. Because now we see And the Kristen tweet really showed it, that one structure is not describing what is going on by itself. It's always a dynamics together with the structure, which gives you the function, on which drives the function. So that's the day 2. Day 3 was funny because there were the parts about the predictions of the ligands and ligand binding. And there is still to do, something to do. And it was not that successful. And then the nucleic acids, because we have majority of the structures which is in protein data bank are from the proteins. But for the nucleic acids, we have like 2 orders of magnitude less. And that means you have much smaller amount of the structures to train on. And those structures you have in the database are either really tiny ones, like decamer, of some DNA or RNA, or some RNA loop or something, or you have a large ribosome, which is the one of the largest biomolecules, which is quite nicely structured, but you don't have the information about how the tRNA or mRNA or siRNA are looking, basically, in the database. And those are the structures you are interested in. So the prediction of the structure of those ones seems to be a big deal further. And when people try to use similar approaches as AlphaFold, they were not successful in this round of the CASP. So this is one thing which seems to be interesting. And day 4 was basically EMA. And this one I don't know. Yeah, but it's all of those presentations are here. So if you want to know what's going on and what will be further, it's relatively simple to find it. Anyway, I've put all of the today's slides already on the webpage, so you can find it there. And as soon I will get the slides from tomorrow's lectures, I will be uploading them as well. So I hope especially the tutorials to gather. Want to have the tutorials? Okay, can you send them to me so I can upload them?
Can we distribute on computers from?
Yeah, they will be on the webpage. So it's
like 100 megabytes.
I think that during the listening to the morning lectures, they can download it. Or they can download it to their own computers by itself, of course. Yeah. Okay. If there is nothing else, then that was all for the lectures today. And you won't see me again till Thursday, other than just moving and speaking to you. And now, poster session. So let's time to go, mix and talk to each other. And that's end for the online participation for today. So you can kill the thing.
I use Handouts. Yeah.
I need to add a few masters first.
No jo, já myslím, že to šlo. No. Jo, to se hodili. Ale tohle jsem se moc nenapil. Je dobrý, ale